Hello, welcome to this tutorial on synaptic transmission. Our core concept is once again that neurons communicate using both electrical and chemical signals. Now that we've discussed the means for generating electrical signals, we're ready to talk about how chemical signals are used in neurotransmission. We have a couple of learning objectives for you today. First, I want you to be able to compare and contrast the structural and functional similarities and differences between electrical and chemical synapses. I want you to be able to describe the sequence of events that's responsible for transmission of neural impulses from one neuron to the next via a chemical synapse. I want you to consider that process in more depth and characterize the critical role of calcium in chemical neurotransmission. And lastly, I want you to be able to discuss the mechanisms of action by which Botox, which is a compound found in nature and now produced for various clinical and cosmetic procedures, how this compound affects neurotransmission. Let's begin by considering the general structure and function of electrical and chemical synapses. Electrical synapses, which are illustrated on the left-hand side, function by means of proteins that form channels called gap junctions. And these channels allow for the passage of small ions that carry current directly from the cytoplasm of one neuron to another. Now, in contrast, the way chemical synapses work is via the release of these packages of neurotransmitter into the synaptic cleft. These packages are called synaptic vesicles, and the transmitter itself could be one of a variety of chemical compounds that has to diffuse across the gap between neurons in order to have an effect on the postsynaptic neuron. Now, in order to compare and contrast the general function and structure of electrical and chemical synapses, I would invite you to turn to the very end of the tutorial notes that I've given you and look at the table where I've tried to lay out some general properties that allow us to compare and contrast electrical and chemical synapses. So let's begin with some general functional considerations. Electrical synapses allow for the very rapid communication of electrical signals from one cell to the next. And this is often very useful if it's important to synchronize a local population of cells that might be in contact with one another via these gap junctions. So here's a slightly more detailed rendering of what a gap junction might look like. So what we see here are the close apposition of two membranes, so much so that it allows for the pairs of complementary gap junction proteins to actually line up in registration with one another from across the two cells that are coming together here. So these proteins that form these gap junctions are called connexons, and they aggregate to provide an aqueous channel that allows for a current-carrying molecule to pass directly from the cytoplasm of one neuron to another neuron. So here's another look at what this might look like. So you can see a variety of connexons that are all arrayed here, allowing for ions to pass from one location to another, and with it potentially carrying, let's say, a positive charge, thereby exciting one cell in its neighbor. Now, as I mentioned, this mediates very rapid synaptic effects, so rapid that this is the fastest way that neurons have to communicate one with another. Here in this illustration, we see that virtually the same instance that a presynaptic neuron fires an action potential, the postsynaptic neuron begins to depolarize towards threshold so that it fires an action potential just a fraction of a millisecond after the first action potential in the presynaptic neuron. Now, this can be very useful in a variety of physiological contexts. One is when it's important to synchronize a local population of cells. So look here, we have two neurons that are connected via gap junctions, and when one fires an action potential, not perfectly in sync, but very nearly in sync, there are other cells that fire action potentials. So this is important in certain parts of the central nervous system, such as the hypothalamus, where a certain quantity of a chemical might be released into the bloodstream, and in order to get enough of that chemical into the bloodstream to have a physiological effect, there needs to be synchronization of the whole population of neurons so that they release their hormone, or they're releasing hormone virtually at the same moment in time. Now, if we back up for just a moment, let's look again at the structure here of a typical electrical synapse, and I want you to notice that what's not present. So a few things that are not present: we do not have any vesicles within a presynaptic terminal containing a chemical neurotransmitter. Likewise, we do not have a significant synaptic cleft, that is a gap between the presynaptic neuron and the postsynaptic neuron that must be bridged through some chemical mediator. Rather, we have two membranes that are coming right close together so that the connexon proteins in either membrane can associate with one another, forming the gap junction. Now, these kinds of electrical synapses can form as two membranes come together. They can form from one dendrite to another, from a dendrite to a soma or a cell body, or between two cell bodies, two somata. Now, one other point about the permeant species that can travel from one cell to another through a gap junction. They're often a current-carrying molecule, such as an ion, but they might also be some other small molecule that's important in second messenger signaling within cells, or otherwise important for intercellular communication. Such small molecules that typically pass through these gap junctions can include calcium ions. They can include ATP, adenosine triphosphate. Both of these molecules are important in mediating either tasks that require energy or a second messenger system, such as a calcium-mediated system, that can coordinate the activities between two neurons that are so connected. Now, notice also, even though the arrows here in this figure are illustrated just going in one direction, that the signals may be bidirectional. That is, ions are free to diffuse down their concentration gradients or what other permeable molecule we have in mind, such as calcium or ATP. So there's no directionality to the communication here. It can go in both directions. Now, one other point here: the probability of transmission is very high. That is, when the gap junctions are open, when the connexons form their channels, molecules are simply free to diffuse down their concentration gradients, meaning that an electrical signal generated in one cell will be transmitted to the neighboring cell via the passage of a charge-carrying ion with almost 100% certainty. Okay, now we're ready to consider some general properties, both structural and functional, of a typical chemical synapse. And so if we look again at figure 5.1 from your book, we see on the right-hand side an illustration of a typical chemical synapse. And here, a few obvious morphological features stand out. First of all, there are synaptic vesicles that contain chemical substances that are the neurotransmitters that will be released as those vesicles fuse with the presynaptic terminal. There is a distinct cleft between the pre and the postsynaptic element, and that cleft is an extracellular space through which the neurotransmitter must diffuse. Now, the chemical synapse, this is the principal type of synapse we find in the adult nervous system. I should have made this point earlier. Electrical synapses are much more common throughout the nervous system as the nervous system is first being constructed in embryogenesis. But with the exception of a few special populations, such as in the hypothalamus, where we have those hormone-secreting cells, and perhaps among some inhibitory cell networks in the cerebral cortex, electrical synapses are not terribly abundant. The principal transfer of information that happens throughout the central nervous system is mediated via chemical synapses. So those are the synapses that we're going to spend the most time over the next couple of tutorials thinking about their function. So, as I mentioned, there's a distinct synaptic cleft, so it's not possible then to have connexon proteins forming gap junctions or aqueous pores through which ions can pass directly from one cell to the next. Rather, there's a cleft. So there's a gap that must be mediated with some chemical message that diffuses across that synaptic cleft. Now, in addition to the presence of neurotransmitters, of course, we also have receptors for those neurotransmitters that are found in the postsynaptic membrane, another distinctive feature of the chemical synapse. Chemical synapses typically are from an axon to a dendrite, or more specifically to a small protuberance called a dendritic spine, but chemical synapses can be from an axon terminal directly to a cell body. They can even be from one axon terminal to another. So quite a lot of morphological variations can be seen with chemical synapses as well as with electrical synapses. Now notice that current does not flow directly from one cell to the next with a chemical synapse. There's this mediator, this neurotransmitter. So once a chemical neurotransmitter has bound to its receptor, something has to happen for there to be the generation of current flow and another action potential in that postsynaptic cell. So we're going to take some time to think through exactly how that happens over the next couple of tutorials. Now, with respect to timing, all synapses are fast, but clearly the electrical synapse is designed for the most rapid, most efficient form of transmission. So there's virtually no synaptic delay when it comes to an electrical synapse, but for a chemical synapse, all of these processes that we see outlined here to the right of this figure, they all take some time to unfold. So relative to the electrical synapse, the chemical synapse is about maybe 4 or 5 times slower. Still fast, still maybe about a half millisecond or so delay at each synaptic junction, but still not nearly as fast as the electrical synapse. So chemical synapses are going to be less effective at synchronizing local populations of neurons than the electrical synapse. A few other points to make. The chemical synapse is unidirectional. That is, there's release of transmitter from the presynaptic terminal and it binds to receptors on the postsynaptic terminal. Now, for the aficionados, you'll recognize that there are presynaptic receptors for transmitters, and there might be retrograde signals given off by the postsynaptic cell. But for the most part, information flow is from presynaptic terminal to postsynaptic terminal, unlike the bidirectional transmission that can clearly happen at the electrical synapse. And lastly, before we leave our table, it's worth noting that the probability of transmission at the chemical synapse is surprisingly low. For synapses in the brain, typically, the probability of there being a postsynaptic spike given a presynaptic spike is on average somewhere on the order of 5 to 10%. Of course, it varies depending upon the synaptic connection in question. But for the most part, chemical synapses in the brain are very weak and they're very unreliable. And there's actually a pretty good reason as to why that might be adaptive, which we'll talk about when we discuss synaptic plasticity. Okay, at this point, I think we're ready for a closer look at the structure of a typical chemical synapse. So let's jump back to the first page of your tutorial notes and Roman numeral 3 if you're following along, and let's have a closer look at a chemical synapse. Now, figure 5.3 is from your textbook, and if you've got the textbook, I would highly commend this figure to you. I think it's a very useful figure for spending some time and studying and maybe even drawing some of this out for yourself, because it will really help you understand some of the fascinating cell biology and molecular biology that's responsible for chemical synaptic transmission. So let's follow this figure, and we can follow along in our notes, which are designed to go right along with what we see in the figure. So let's consider the sequence of events that unfolds as an action potential leads to the release of neurotransmitter and the generation of a postsynaptic signal. This process begins in the steady state, and what we find there in the presynaptic terminal are a variety of organelles that are involved in the production of energy, the production of the membrane that's needed to package up neurotransmitters into vesicles, and then the machinery itself that manages the flow of vesicles and ultimately their fusion with the presynaptic terminal. So we find a variety of important proteins and structures that are present within this synaptic terminal. And I'll show you that in more detail in a few minutes, and we'll just highlight a few of the important members of that cellular machinery as we go along. Now, I want you to notice what we have here in this presynaptic terminal with respect to our synaptic vesicles. So there are some vesicles that seem to be floating freely in that presynaptic terminal, and then there are others that seem to be attached to the presynaptic terminal membrane. So there's a pool of vesicles that are free, and there are a pool of vesicles that are attached. Those that are attached we call docked. And so these vessels that are docked are localized near what we call the active zone, which is going to be the site of fusion of those vesicles with the plasma membrane. Those docked vesicles are the ones that are docked and ready to go when an action potential arrives. Okay, so sure enough, an action potential does indeed arrive at the presynaptic terminal. So this is step number 2 now. And so a wave of depolarization passively diffuses along this pre-terminal membrane until we encounter a voltage-gated calcium channel. So near the active zones, there are numerous voltage-gated calcium channels that are present, and as the wave of depolarization hits, those channels open, and now calcium rushes into the presynaptic terminal. So step number 4 here, and this is because there is a steep concentration gradient with more calcium outside in the extracellular fluid than inside that presynaptic terminal. The influx of calcium is really key, because as calcium rushes in to that presynaptic terminal, that is the key event that happens in space and time at the active zone and in time following the wave of depolarization that hits the active zone. Calcium is what triggers the fusion event that leads to the fusion of the synaptic vesicle with the presynaptic terminal and the release of neurotransmitter. Okay, so calcium rushes into the cell, and it causes these vesicles to begin to fuse with the presynaptic terminal membrane. That allows their neurotransmitter to be released into the synaptic cleft via exocytosis. Very similar process happens with all other kinds of secretory cells in the body. So the axon terminal is not particularly unique in that regard, but it certainly is specialized to release neurotransmitter via synaptic fusion events very rapidly and very efficiently. Now, once the neurotransmitter is released into the synaptic cleft, then that neurotransmitter diffuses where it can interact with a number of postsynaptic receptors. Some of that neurotransmitter, of course, is just free to diffuse away, which it does. But the interaction of the neurotransmitter with the postsynaptic receptor is what triggers the transfer of the information from presynaptic terminal to postsynaptic process. If the neurotransmitter binds to a receptor that happens to open a cation selective ion channel, then cations can enter that presynaptic terminal. That can lead to the depolarization of that postsynaptic site. If this happens over many hundreds or potentially thousands of terminals on a cell, then that very likely will summate and lead to the depolarization to threshold of the postsynaptic neuron, and an action potential might be generated. Without such massive summation, our typical synapse in the brain is not likely to depolarize the postsynaptic neuron to action potential generation. But nevertheless, this is the general process. Transmitter binds to receptor. Receptor can open a cation selective channel. Cations can flow into the cell, leading to their depolarization. But that's not the only thing that can happen. Neurotransmitters can bind to channels that can allow for negatively charged ions, anions, to flow into the cell, such as a chloride ion. That might lead to the subthreshold depolarization of the cell or the hyperpolarization, depending upon what the reversal potential is of the ion channel in question. There are also receptors that mediate channel closing events. So that can either lead to depolarization or hyperpolarization, depending upon the permeability of that particular channel. So there's a lot of activities that can occur when a neurotransmitter binds to its postsynaptic receptor. We'll talk about them in more detail over the next couple of tutorials. All right, so what happens to the neurotransmitter after it's been released? Well, I mentioned neurotransmitter is free to diffuse away. For some neurotransmitters at some kinds of synapses, there may actually be an enzyme that degrades that transmitter. In other systems, the glial cell, typically the astrocyte, which is going to be present around synaptic junctions, can actually take up the neurotransmitter, or perhaps a metabolite of that neurotransmitter, and begin a process that will recycle that substance back to the presynaptic terminal. And then finally, the last thing that typically happens is that the membrane from the synaptic vesicle is retrieved and recycled so that the specialized proteins associated with that membrane can be reused and the membrane itself can be recaptured. Obviously, if a synapse were to simply fuse with all of its vesicles over time, the surface area of that synaptic terminal would continue to expand, and that would not be consistent with the viability of that synaptic structure. So it makes great sense that there's some kind of process that would recycle that synaptic membrane, and indeed there is. Just a word about the timing of all of these events. So chemical synapses are slower than electrical synapses, but chemical synapses are still quite fast, and it really is remarkable how quickly these activities can unfold. For example, the process of forming a synaptic vesicle and filling it with neurotransmitter, docking it, having it ready to go at an active zone might take place in less than a minute. The actual fusion events where the membrane fuses, causing a release of neurotransmitter, can happen in about a millisecond or less. And then finally, the recycling of that membrane back into an endosomal compartment can take place in less than a minute. So very quickly, this process can unfold at every synaptic terminal in our brain. Now, let's consider the critical role of calcium in the fusion of presynaptic vesicles with the presynaptic terminal membrane. So I'd like for us again to consider some evidence that supports the important role of calcium in the process of vesicles fusing with the presynaptic terminal. These data come once again from our favorite animal model for these kinds of studies, the squid, where there are some really huge synapses that allow experiments to be done like can't be done in nearly any other known system. And what's being done here is a presynaptic neuron is being stuck with microelectrodes that allow for clamping the voltage or for injecting current. And there's a postsynaptic process, postsynaptic neuron, from which the postsynaptic membrane potential is going to be recorded. And so what's being demonstrated here in these data is the important role of calcium. So if the presynaptic terminal is depolarized, which is what we see here, there's an inward current, and that inward current is carried by calcium. So basically calcium is rushing into the presynaptic terminal as voltage-gated calcium channels open. That leads to an action potential in the postsynaptic neuron. Okay? Now, this presynaptic terminal is large enough that the scientists can actually infuse a compound that blocks calcium channels, and when that's done, the same kind of depolarization of the presynaptic membrane fails to result in the opening of these voltage-gated calcium channels. So there's pretty much no change and no action potential. So without calcium in the presynaptic terminal, we do not record an action potential in the postsynaptic process. Likewise, a series of experiments were done that further this theme, which illustrate that calcium is both necessary and sufficient to trigger vesicle fusion and the release of neurotransmitter. So now, using the same preparation, it's possible to inject calcium directly into the presynaptic terminal. And when you do that, you can measure a depolarization of the postsynaptic neuron. So this suggests that infusion of calcium into the presynaptic terminal leads to some vesicle fusion events, the release of neurotransmitter, the opening of cation selective channels in the postsynaptic membrane, and the depolarization of that cell. Here again is further evidence for the importance of calcium. In the control experiment, the presynaptic membrane is depolarized such that an action potential is generated. About a millisecond later, an action potential is recorded in the postsynaptic cell. But if a calcium chelator is injected into the presynaptic cell, this is a compound that is going to prevent a sudden rise of intracellular calcium. So now we're not blocking the calcium channel, we're just sucking up the calcium that enters the presynaptic terminal. So even though we have a nice, robust action potential in the presynaptic neuron, there's only a very modest subthreshold depolarization seen in the postsynaptic cell. So this experiment suggests that a sudden rise of calcium in the presynaptic terminal is critical for the release of neurotransmitter and the generation of an action potential in the postsynaptic cell. Okay, well, so far we haven't really discussed how all this comes about. And to begin that discussion, we need to consider some of the molecules that are affiliated with the synaptic vesicle membrane as well as the membrane at the presynaptic terminal. So here's a rather fanciful look at a synaptic vesicle, and what we see is an artist's rendition, now a couple of years old, but still it just conveys the point of the complexity from a molecular perspective of the surface of the vesicular membrane. And this complexity reflects the precise means by which the presynaptic terminal regulates the trafficking of synaptic vesicles and controls their fusion with the presynaptic terminal. So I show you this just to make the point that there are dozens of molecules in the vesicular membrane that are important for regulating the activity at a chemical synapse. One of these molecules is particularly important, and I will highlight that for you. It's a protein called synaptotagmin, and here it is illustrated here. So synaptotagmin appears to be the molecule in the vesicular membrane that binds to calcium. So as calcium rushes in to that presynaptic terminal, synaptotagmin seems to be the calcium sensor that interacts with calcium and eventually sets into motion the events that quickly unfold, leading to the fusion of the membrane of the vesicle and the presynaptic terminal. The way that this happens is through the activation of a set of proteins, some of which are found in the synaptic vesicle membrane. Others are found in the membrane of the presynaptic plasma membrane. Together, these proteins form something called the SNARE complex. So SNARE is actually an acronym. You don't need to be concerned with knowing that acronym, but it's helpful to use this acronym because it reminds you as to the function of this complex of proteins. So the SNARE complex is involved in snaring the vesicle membrane to the cytoplasmic face of the presynaptic terminal. This is, in fact, the docking process. So as these membranes approach one another, there is an interaction among these proteins. Basically, these proteins, such as synaptobrevin, SNAP25, and some others, they associate with one another and perhaps form some sort of helical interaction that allows the synaptic vesicle to be docked to the presynaptic terminal. Now notice the proximity of the SNARE complex to synaptotagmin. Synaptotagmin, again, is the molecule that binds calcium ions as calcium enters this presynaptic terminal with the arrival of depolarization. So let's sort of step through that process and see how this works. So initially we have proteins affiliated with the vesicle membrane. We have synaptobrevin, SNAP25, and synaptotagmin. But not yet do we have significant depolarization arriving. So the snare complex begins to dock a vesicle membrane, and this happens in a calcium-independent fashion. This allows a vesicle to be docked even without an inrush yet of calcium into the presynaptic terminal. So when the snare complex forms, we can say that these membranes have been pulled together and this vesicle is now docked. So this is a picture of what a docked vesicle looks like. The snare complex has formed. This vesicle is primed and ready for action with the arrival of a wave of depolarization. So once that happens, voltage-gated calcium channels open, calcium rushes into the presynaptic terminal, and now calcium binds to synaptotagmin. And the interaction of calcium and synaptotagmin leads to a series of conformational changes in the snare complex that tugs these two membranes together. And as that happens, then the membranes will fuse, and neurotransmitter now is free to diffuse out of the synaptic vesicle through what's called a fusion pore that forms as the vesicle membrane and the presynaptic terminal unite. Well, now that the neurotransmitter is diffused out of the synaptic vesicle, what's left is the retrieval of the membrane from the synaptic vesicle. And this involves a variety of proteins, some of which are illustrated here in this slide. I would highlight just a couple of them for you. One that's very important is this protein called clathrin. So clathrin coats the vesicle membrane, and with interactions with other proteins, such as this dynamin protein, we have the retrieval of those membranes. And here's a picture of what this looks like. So the clathrin molecule has an interesting geometrical shape. We see it up here to the upper left. It forms a structure called a triskelion, and that triskelion then can associate with other molecules and essentially form this buckyball-like ring around the vesicular membrane. And so as these clathrin triskelia begin to form, then the vesicle membrane is pulled back away from the presynaptic terminal. At some point in here, dynamin gets involved and appears to nip these vesicle membranes, allowing for a completely coated vesicle membrane to now be retrieved, and via interactions with actin filaments and a variety of other proteins, this vesicle membrane is retrieved, the clathrin coat is removed, and the vesicle is now ready for repackaging with neurotransmitter without loss of all of those specialized proteins that are associated with this vesicle, like synaptotagmin and the snare complex, that are important for the function of this membrane as it's recycled and repackaged with more neurotransmitter. Now, I'd like to conclude this tutorial by thinking about the activity of a toxin that's found in nature from the Clostridium family of bacteria, and it's over the last couple of decades or so has been used relatively widely in clinical practice, and also, unfortunately, been somewhat abused perhaps in the cosmetic industry. And of course what I'm speaking of is botulinum toxin, otherwise known as Botox. So Botox can be used in the therapeutic setting to reduce muscle tension, to reduce muscle spasticity, perhaps to quiet a twitching muscle. These are all very good and appropriate uses of Botox. Botox can also be used cosmetically to try to reduce muscular activity that might give rise to wrinkles or furrows in the skin. And for a variety of reasons, Botox has become a fairly widely used drug in our time. So how does it work? Well, the way Botox works is by cleaving the snare complex. So Botox is a protease. And it is a protease that disrupts the SNARE complex in one of several sites, depending upon exactly what formulation of botulinum toxin we're speaking of. One of the most common and widely used forms of Botox is Botox A, and we now know exactly where the cleavage site for Botox A is within the SNARE complex, and a variety of other Botox formulations have their own cleavage sites on one member or another of the SNARE complex. So you can imagine why this works so well. So if the SNARE complex is cleaved, then no matter how much calcium rushes into the presynaptic terminal, that vesicle is very unlikely to fuse on its own. The SNARE complex is what facilitates bringing together these membranes and twisting and turning, resulting in the fusion of the vesicular membrane with the presynaptic terminal. So without the SNARE complex to pull those membranes together, we're very unlikely to have a fusion event, and therefore we're very unlikely to release neurotransmitter at that synaptic terminal. So Botox is a very powerful molecular tool that can be used to silence a chemical synapse. Okay, well, we've come to the end of the tutorial, and I would encourage you to view one of the animations that will review these processes that are involved in synaptic transmission. It's animation 5.1. You can follow the hyperlink at the end of the handout or navigate there via our website. Next time, we'll talk about neurotransmitters, and after that, the receptors for neurotransmitters. So, I'll see you then. Welcome back to my home. I'm out in my yard on a kind of a muggy gray day. It's been raining, so the air feels a little bit like it might start raining on me at any moment now, so we'll see what happens. But let's keep going. So we're here today to talk about neurotransmitters. So this reflects our second core concept in the field of neuroscience. Again, continuing on in our thinking about the important means by which neurons communicate using electrical and chemical signals. So now we're going to focus on what exactly those chemical signals are. So our learning objectives today are that I want you to be able to name the major small molecule neurotransmitters and neuropeptides in the central nervous system and briefly state the functions of each. And I want you to think with me about the factors that determine the effect of neurotransmitters on postsynaptic neurons. All right. Well, let's begin by reviewing what we talked about last time about the organization of chemical synaptic transmission. And this slide gives you again an overview of the processes that are involved in chemical synaptic transmission, beginning with the steady state, where we find in the presynaptic terminal a variety of organelles, and of course our presynaptic vesicles that are loaded up with neurotransmitter ready to go. And what happens then with the arrival of an action potential is a wave of depolarization spreads along the pre-terminal membrane, which eventually reaches voltage-gated calcium channels. And those channels open, calcium rushes in, which is the key triggering event, which leads to the fusion of the presynaptic terminal membrane and the membrane of the synaptic vesicle. So that happens as the SNARE complex, which is present out here, twists and tugs and pulls those membranes together once calcium associates with synaptotagmin. And as those membranes begin to fuse, they form what we call a fusion pore, and that allows for neurotransmitters then to diffuse out of the synaptic vesicle into the synaptic cleft. And from there, the neurotransmitters can associate with receptors for those neurotransmitters in the postsynaptic side of the synapse. And as receptors and transmitters interact, typically ion channels open and close. That could lead to the generation of a wave of depolarization in that postsynaptic process, and maybe even with enough summation of inputs that are doing the same thing at the same time, there could be an action potential generated in that postsynaptic cell. Meanwhile, the membrane of the vesicle is recycled back for further activity. And this involves the coating of the vesicle membrane with a protein called clathrin, and the association of clathrin with other molecules that facilitate the pinching off of that vesicle membrane, and then it's trafficking back into the pool of vesicles that are ready to be filled again with their neurotransmitter and re-enter this vesicle cycle. Okay, well, let's let that suffice as a brief review of chemical synaptic transmission. Again, I would refer you to the animation associated with the textbook Animation 5.2 on chemical synaptic transmission, which will review all of these processes in a nice, short, succinct presentation. All right, well, what we're here to talk about today is neurotransmitters. And neurotransmitters come in two broad kinds of categories based on their structure. And these two categories of neurotransmitters interact with different kinds of receptors. So we have two broad classes of neurotransmitters based on the size of the molecules. One category we call small molecule neurotransmitters for obvious reasons. And then to contrast those small molecule transmitters, we have neurotransmitters that are actually small peptides. And so these peptides, of course, are comprised of amino acids that are covalently bound to one another, and sometimes these peptides can be quite sizable. So small molecule transmitters and peptide transmitters. So these two classes of transmitter bind to two classes of receptor. One class of receptor we call ionotropic receptors. These are receptors where the receptor site is actually part of an ion channel, and that ion channel can open or close when the transmitter binds to the receptor. So these ion channels are called ligand-gated ion channels, with the ligand being the neurotransmitter. The other type of receptor that we'll consider today are called metabotropic receptors, and these receptors activate second messenger system. So there are metabolic steps involved in mediating the effect of the neurotransmitter once it binds to its receptor. Now, our small molecule neurotransmitters can actually interact with both of these classes of receptors, both the ionotropic receptors and the metabotropic receptors. So right away, I want to emphasize to you that if we want to understand the activity of a neurotransmitter, we need to know something about the receptors that it interacts with. And the activities of neurotransmitters, even on the very same cells, can be remarkably diverse, depending upon what kinds of receptors are expressed on that neuron. Our peptide neurotransmitters, as far as we know, as far as I know, only interact with metabotropic receptors. So, as we'll come to see, these metabotropic receptors mediate slower and potentially longer lasting effects in postsynaptic neurons. So we associate our small molecule transmitters primarily with quick actions, and our peptide transmitters, generally speaking, with more long-lasting and slowly evolving actions. Okay, well, let's begin with a survey of some important small molecule transmitters. Now, for those of you following along in the reading here in Chapter 6 goes into more depth than what we'll cover. So I want you to focus on the level of knowledge that I've given you in the tutorial notes and the level of my discussion with you all today. So let's begin by thinking about these small molecule transmitters and get a broad functional sense of what these molecules are. So many of them are amino acids. Some of the amino acid transmitters that are important in brain and spinal cord function are shown here. They include amino acids such as glutamate, potentially aspartate, certainly gamma-aminobutyric acid, or GABA for short, and glycine. So these molecules mediate rapid synaptic effects when they interact with their ionotropic receptors. As I mentioned, they may also interact with metabotropic receptors. So the small molecule transmitters can also have longer lasting effects when those particular kinds of receptors are bound. Now, here we're illustrating some of the important small molecule transmitters that you'll want to know about, and perhaps the most important of them all is glutamate. So glutamate is typically considered the major excitatory neurotransmitter in the central nervous system. And as you see here, it is an amino acid, so short carbon skeleton with the amino group attached to the side opposite the carboxyl group. So glutamate is involved in various aspects of cellular metabolism, but it also is concentrated in synaptic vesicles at the terminals of neurons that use glutamate as a neurotransmitter. Now, this is such an important molecule that we've prepared some animations that talk about the processing of this molecule as a neurotransmitter, so I'd encourage you to pause just a minute and view animation 6.2 on the website that supports our textbook. You can navigate there yourself or click on the hyperlink in your tutorial handout. Well, next, let's consider acetylcholine. So we're deviating just a little bit from the flow of the handout, but that's okay. Acetylcholine is another small molecule transmitter. It's not an amino acid, but it is a small molecule. And acetylcholine has very important activity both within the central nervous system and outside of the central nervous system. Outside of the CNS, it's the major excitatory neurotransmitter of our somatic motor neurons. So this is the neurotransmitter that's released on muscle fiber, leading to the contraction of muscles. It's also a major excitatory neurotransmitter in autonomic ganglia, and it's also the neurotransmitter of many postganglionic parasympathetic fibers. Within the brain itself, it seems to have an important modulatory role that may be important in various cognitive functions such as attention. Just as an example of how one such small molecule neurotransmitter might be processed, let's take a closer look at a cholinergic synapse. And so we have this illustrated here in figure 6.2. And so within the presynaptic compartment, we've got the metabolic machinery that's necessary for producing acetylcholine. In this case, acetylcholine is synthesized by an enzyme called choline acetyltransferase from acetyl-CoA, one of the products of oxidative metabolism, and choline, a dietary compound that's important for various aspects of nutrition and brain health. Well, acetylcholine is synthesized in the presynaptic terminal, packaged into synaptic vesicles, and then released in a calcium-dependent fashion. When acetylcholine is in the synaptic cleft, it can interact with postsynaptic receptors, which can be either of the ionotropic or metabotropic variety. And once acetylcholine has been released into the synaptic cleft, it can be degraded by an enzyme called acetylcholinesterase. And when that happens, the choline portion of the molecule is taken back up into the presynaptic terminal and recycled. To learn more about the function of a typical cholinergic synapse and how all this works, you might want to view animation 6.1 on our website. Now, before moving on to the biogenic amines, let me just back up a moment and talk about the other important amino acid neurotransmitters that we skipped over, namely the two at the bottom here, GABA and glycine. So GABA is short for gamma aminobutyric acid, and it's a metabolite of glutamate. So glutamate is in the biosynthetic pathway for GABA. Glycine is a different amino acid. Now, both of these amino acids typically have inhibitory activity in the mature nervous system. We'll come back and say quite a bit more about GABA as we go, but for now, let me just highlight that GABA is the single most important transmitter in the brain that mediates synaptic inhibition. Glycine is a very important neurotransmitter, especially in the spinal cord, where it likewise mediates synaptic inhibition. Glycine also plays an important role at synapses that release glutamate, where glycine serves as a co-transmitter whose binding is important for the activity of one type of receptor for glutamate. Okay, so now we're ready to move on to another class of small molecule neurotransmitter. These are the biogenic amines, and these include molecules such as dopamine, norepinephrine, epinephrine, serotonin, otherwise known as indolamine, and histamine, based on yet a different type of carbon ring structure. So these are all amine molecules that are linked to some kind of a ring type of structure. And they all have biological activity in the nervous system that tends to modulate the activity of neural circuits. So unlike molecules like glutamate and GABA that mediate rapid synaptic effects that appear to convey information as information flows in neural circuits, the biogenic amines rather modulate the way those circuits function. And we'll talk more about them when we see them in context in structures such as the basal ganglia of the forebrain. But it's worth emphasizing here that these substances are very important in motivation and reward systems, such as the compound dopamine. Dopamine is also critical for modulating movement in circuits such as the basal ganglia, and it's involved in activation of circuits in the prefrontal cortex. This is very important in helping to mediate various aspects of cognition. Likewise, norepinephrine is also very important in cognitive functioning. It seems to be part of what it means to pay attention, is to have a heightened release of norepinephrine, along with other molecules such as acetylcholine. Another important small molecule transmitter among the biogenic amines is serotonin. And together with dopamine, serotonin and dopamine together seem to account for many of the diagnostic categories that we encounter in psychiatry, in the human behavioral sciences. In fact, most of the drugs that are prescribed to treat psychiatric illness act on modulating the activities of dopamine and serotonin synapses in the brain. Let's look anatomically for just a moment and consider the source of some of these biogenic amine transmitters, beginning with dopamine. Well, I mentioned some of the places where dopamine is released. Dopamine is derived from cells that have their cell bodies here in the midbrain, which is at the very top of the brainstem, in a collection of compact cells known as the substantia nigra pars compacta. And right adjacent to that is a region called the ventral tegmental area. Here we have cells that have their somata here, and they send axons to important parts of the brain, such as the basal ganglia, specifically the striatum, which is part of the basal ganglia. And from the ventral tegmental area, projections into medial temporal lobe structures, such as the amygdala, as well as to this ventral and medial aspect of the prefrontal cortex. So dopamine is a very important neuromodulator in all of these places. Here, just to highlight a little bit of neuroanatomy, we have a cross-section through the forebrain, as illustrated in the upper right of this slide, shows you about where we're looking. And I want to draw your attention to the center of this slice. So you'll notice labeled number 11 here, we have this dark substance which sits in the midbrain. And that dark substance is, in fact, the substantia nigra, which of course is Latin for dark substance. So here we've got the cell bodies of the neurons that synthesize and release dopamine at some distance at their axon terminals up in the basal ganglia and in the cortex. And in the medial part of this region where we have dark substance, that's our ventral tegmental area. And that too is a place where the cell bodies of these neurons reside that send their axons up into structures associated with the limbic forebrain and release dopamine there. Well, norepinephrine is another important small molecule neurotransmitter, And norepinephrine is derived from cells who have their home in the brainstem, but now in the dorsal part of the pons, in a structure called the locus ceruleus. The locus ceruleus is another place where we find black substance, or actually the locus ceruleus means blue spot. If we are lucky enough to cut a section through the tegmentum of the pons just right, we might see these two little spots that look sort of a deepish grayish blue color. Those are the locations of where we find our cell bodies of neurons that are producing norepinephrine and releasing it in various places in the brain, really just about everywhere in the brain, and even down into the spinal cord do these cells grow their axons. So all of these biogenic amines have in common a limited number of cells whose cell bodies are in the brainstem that grow axons that branch and grow profusely throughout various parts of the forebrain, hindbrain, and even the spinal cord. One last example is the neurotransmitter serotonin. So serotonin comes from a group of cells that are found in a couple of different places in the brainstem. They tend to be found right along the midline in a group of nuclei that we call the seam nuclei, or the raphe nuclei. Raphe means seam, so right along a seam where the two sides of the brainstem come together. That's where we find our cell bodies that release serotonin. And as I mentioned, serotonin is a very important neuromodulator. Serotonin appears to be a molecule that, when out of proper physiological balance, can lead to various affective disorders. Many of our drugs that we use to restore normal health with respect to affective disorder act by selectively blocking the reuptake of serotonin, thereby increasing the synaptic concentrations of that drug in important brain structures. Okay, well, there are more animations to view. If you care to see some summary of some of the biochemistry and some of the molecular biology regarding the function of these biogenic amine synaptic systems, I would encourage you to view animations 6.3 and 6.5 that review the organization of dopamine and serotonin synapses. Okay, at this point, I think we're ready to switch gears just a bit and talk about the neuropeptide neurotransmitters. So as I mentioned, the neuropeptide transmitters are larger molecules. They're made of a number of amino acids that are covalently bonded to small peptides. Sometimes they can be rather short, or sometimes they can be of considerable size. So, as you might imagine, these molecules must be handled somewhat differently in the presynaptic terminal, and indeed they are. And we'll come back to that in just a minute. But before we talk more about how they're handled in the presynaptic terminal, let's just make a few points about their physiology. So the neuropeptides mediate slower and more long-lasting effects than most of our small molecule transmitters, and that's because the neuropeptides seem to interact with metabotropic types of receptors. And as we'll discuss, these are receptors that require cellular metabolism in order to mediate their function, and all of that takes time. So the neuropeptides mediate slower and more long-lasting synaptic effects, some of which can even go on for hours to days, depending upon the downstream effects of the metabolic pathways that are activated. Some of the important neurotransmitters in this category include what's shown here in this slide, the enkephalins. So there are a number of different enkephalins that are important. The enkephalins are opioids that have analgesic effects in the brain, and so these bind to receptors in various places, including the spinal cord. Other important peptide transmitters are substance P, which is very important in modulating the transmission of pain signals in the dorsal horn of the spinal cord. And many of our hypothalamic releasing hormones that are secreted into the portal circulation from the hypothalamus to the anterior pituitary are neuropeptides, as are the hormones that are released directly in the posterior pituitary into the bloodstream from hypothalamic neurons that send their axons into the neurohypophysis. So these hypothalamic hormones are other examples of neuropeptides. And lastly, let's consider the activities of some neurotransmitters that are still relatively poorly understood. And for that reason, we call them unconventional neurotransmitters, not because they're necessarily so different from our small molecules and our peptides. In some cases they are, but mainly because we don't yet have a full understanding of their mechanisms of action. And so let's begin by talking about the purinergic neurotransmitters. So these include our purines, such as adenosine triphosphate. So you're all familiar with this molecule from your studies of cellular metabolism. You know that ATP is the energy currency for cells. And what you may not have known is that it actually has some biological activity at synapses in the nervous system. So ATP can be co-released with other small molecule neurotransmitters. Once it's in the extracellular space, ATP is typically metabolized into adenosine, and adenosine itself can have its own biological activity. Adenosine tends to bind to various receptors, and these receptors have various functions in different kinds of brain circuits. One important circuit where adenosine is important is in the hypothalamus. And adenosine appears to build up throughout our day and appears to be an important trigger that leads to drowsiness. So the more our cells in the hypothalamus are consuming energy, the more adenosine builds up in the extracellular fluids. That seems to be something like an index of our state of fatigue and the degree to which we require sleep. Well, one thing many of us do, I myself am guilty of this, is that we fight this system by consuming caffeinated beverages. And what these caffeinated beverages do is they block adenosine receptors. So the xanthines, including caffeine and theophylline, they are blocking the receptors that are mediating this natural sense of drowsiness that will, we think, in some way have a restorative function on brain circuits. Another of these so-called unconventional neurotransmitters that's fairly recently been discovered are a series of molecules that are derived from membrane lipids called endocannabinoids. These molecules are hydrophobic, so when synthesized they can freely diffuse across plasma membranes, and they bind to receptors that are also bound by the psychoactive components of cannabis, hence their name endocannabinoids. So these include some very interesting receptors such as the CB1 receptor, which is present in high concentrations in structures associated with the brain's reward system. It's also present throughout the cerebral cortex and in the cerebellum, in the basal ganglia. So lots of places where we think that the drug of abuse, the cannabinoids derived from cannabis, have their effects on cognition and sensory and motor function. So these endocannabinoids seem to have some interesting physiological roles in modulating plasticity, especially plasticity at inhibitory synapses. And you can read more about that in the textbook if you like, and figure 6.19. We'll come back to this when we talk about mechanisms of synaptic plasticity in a future tutorial. One final neurotransmitter to discuss is nitric oxide. So this truly is an unconventional neurotransmitter in that it's a gas. It's not a solute that can be dissolved in an aqueous medium. It's not a peptide, but rather it's a gas. And nitric oxide is produced from the metabolism of the amino acid arginine, as is illustrated here in this pathway. So arginine is processed by an enzyme called nitric oxide synthase, and nitric oxide in a gaseous form is produced, as is citrulline, and that nitric oxide then is free to diffuse across the plasma membrane, or it can interact with target structures, maybe within the cell where the gas was synthesized. Here in this particular illustration, we're showing the activation of a guanylyl cyclase enzyme, which results in the production of cyclic GMP, one of the important second messenger systems we have within the brain. Now, one of the interesting activities of nitric oxide involves its ability to diffuse freely from, let's say, a postsynaptic cell to a presynaptic cell. So there's ongoing interest in the idea that nitric oxide might be some kind of a retrograde messenger that allows from signals to travel back from a postsynaptic cell to a presynaptic site. Of some interest is the activities of nitric oxide as a vasodilator. And so this is a function that is performed in a variety of tissues, and that activity is mediated by a guanylyl cyclase pathway. And perhaps it's of interest to some of you that Viagra and other related drugs that are used to affect erectile dysfunction function by affecting nitric oxide mediated vasodilation. Similar pathways, however, are found in a variety of structures within the brain and elsewhere in the nervous system. Hence, these drugs can often have side effects, often involve sensory processing. And we'll see why when we talk about the mechanisms of phototransduction, and we'll discover an important role for the guanylyl cyclase pathway within the photoreceptors of our retinas. Okay, well, let's look now at a side-by-side comparison of the processing of small molecule transmitters and peptide transmitters. So I would encourage you to have a look at the table that I've prepared for you in in page 3 of your handout, which talks about the synthesis, the packaging and release, and the removal of neurotransmitter at synapses that use small molecules and peptide transmitters. So with respect to the synthesis, small molecules are synthesized typically in the presynaptic terminal, where there are enzymes that are present that can create the active neurotransmitter right there in the presynaptic terminal. For peptide neurotransmitters, the synthetic pathway is primarily in the cell body, where the peptide is processed like all other peptides through the endoplasmic reticulum, the Golgi apparatus, where various kinds of post-translational modifications may be performed. Meanwhile, this peptide can be packaged into a vesicle of some sort and then transported down the axon. And along the way, further modifications or perhaps cleavages of that peptide can occur as the initial product of the transcript is then processed and prepared for its active function in the presynaptic terminal. A couple of other differences to note. Small molecule transmitters typically are packaged into vesicles that then are concentrated right along the active zones at the presynaptic membrane, whereas peptide transmitters are often found in vesicles that are diffusing around the presynaptic terminal further away from that active zone. So consequently, the vesicles that contain the neuropeptides, which typically are much larger than those that contain the small molecules, are further removed from those active sites and therefore require a larger influx of calcium in order to trigger calcium-mediated fusion events. So with a relatively modest depolarization of the presynaptic terminal, we're likely to only see the calcium-mediated fusion of small vesicles that release the small molecule transmitters. It's only with a very large depolarization that the presynaptic terminal is likely to release much peptide neurotransmitter, because in order for that to happen, sites well away from our typical high-efficiency active site are engaged, and those larger vesicles then fuse and release peptide neurotransmitters, but only when that presynaptic terminal is flooded with calcium. So this might be one physiological way of holding in reserve the peptide neurotransmitters only when there is intense depolarization of the presynaptic terminal. And lastly, considering the removal of the neurotransmitter once it's been released, the small molecule transmitters have a variety of systems. The transmitter itself might be degraded, and precursor substances might be then taken back up into the presynaptic terminal, or they may run through a glial cell where they may be in some storage form before being returned from a glial compartment to the presynaptic terminal. Of course, the transmitter can simply diffuse away as well. For a peptide neurotransmitter, these substances likewise are free to diffuse away, but there are also proteases that are found here in the synaptic cleft that can cleave these peptide neurotransmitters and degrade them right at their region of release in and around the synaptic cleft. So let's also take a few minutes to consider a typical glutamatergic synapse in the brain. So as I've already mentioned, glutamate's one of the most important small molecule neurotransmitters that we have. It's widely considered to be the most important small molecule for mediating fast excitatory events in the central nervous system. So in a typical presynaptic terminal, we have an important enzyme called glutaminase. And this enzyme converts glutamine into glutamate. And glutamate is then concentrated in synaptic vesicles, where it's taken into that vesicle via a transporter called vesicular glutamate uptake transporter, so VGLUT for short. So this synaptic vesicle then, of course, can fuse with the presynaptic terminal. When calcium floods that active zone, the glutamate is free then to diffuse into the synaptic cleft, where it can interact with postsynaptic glutamate receptors. Now, some of this glutamate, of course, diffuses away from the synaptic cleft, where there are excitatory amino acid transporters in the glial cells that surround the synaptic compartment. And of course, the important glial cell that we're talking about here would be the astrocyte. And so the excitatory amino acid transporter takes up the glutamate, where it's converted back to glutamine. So glutamine is, in essence, a storage form of glutamate that can sit in reserve within astrocytes before being recycled as needed back to the presynaptic terminal. So there are other kinds of co-transporters that allow glutamine to cycle back to this presynaptic terminal, where it can once again be hydrolyzed by glutaminase into glutamate, ready for another round of synaptic release. Okay, lastly, let's just make a couple of points here in conclusion. So we've talked about our two classes of neurotransmitters, our small molecules, our peptide transmitters. We've also talked about what we've called some unconventional neurotransmitters. And these neurotransmitters interact with two classes of receptors, our ionotropic receptors, which mediate rapid synaptic events, since the receptor for the ligand is part of an ion channel complex, and there are metabotropic receptors that require the activation of second messenger systems. So they mediate more slowly evolving and potentially longer lasting synaptic effects. Our small molecule transmitters can interact with both kinds of receptors. Our peptide neurotransmitters, as far as we know, only interact with the metabotropic family of receptors. Now, I think from this simple schematic, it should be apparent to you that if we want to understand the postsynaptic activity of a neurotransmitter, we need to know more than simply the identity of that transmitter. It's not enough to know if it's a small molecule or if it's a peptide. So knowing the chemical structure itself is not sufficient to understand the physiology of the effect mediated by that transmitter. One needs to understand something about the receptors that interact with that neurotransmitter in question. So in order to account for the physiological activity of a chemical neurotransmitter, you need to know the receptor. So in our next tutorial, we're going to talk more about the structure and function of both types of receptors, the ionotropic receptors and the metabotropic receptors. And I think you'll be impressed, as I am, with the diversity of effects that can be activated, even by the very same chemical signal. So Next time we'll talk about neurotransmitter receptors. I'll see you then. Welcome to this tutorial about neurotransmitters. We continue to talk about our core concept about how neurons communicate through both electrical and chemical signals. And last time we spent our tutorial talking about the nature of the chemical signals, the neurotransmitters. Now we're going to talk about the receptors that they interact with. So we have some learning objectives today. I want you to be able to discuss the means by which ligand-gated ion channels affect the membrane potential of postsynaptic neurons. I want you to be able to compare and to contrast the structure and function of the ligand-gated ion channels and the metabotropic receptors, also known as the G protein-coupled receptors. We're going to focus on one very particular ligand-gated ion channel that's very important, especially in the context of learning about neuroplasticity. And I want you to discuss the properties of the NMDA receptor for glutamate and why it's so important for synaptic plasticity. And lastly, I want you to be able to once again recount for the factors that determine the effect of a neurotransmitter on a postsynaptic neuron. Okay, well, let's get started again by reviewing the basic structure and functional properties of a chemical synapse. And so those are recounted for you again in figure 5.3, a very important figure from your textbook. And what we see here is what happens when an action potential invades the presynaptic terminal membrane. So there's a wave of depolarization that causes voltage-gated calcium channels to open. Calcium rushes in to the presynaptic terminal, interacts with synaptotagmin, that causes the snare complex to pull together the membranes of the synaptic vesicle and the presynaptic terminal. That generates a fusion pore, and neurotransmitter then can diffuse out of the synaptic vesicle, where it can interact with neurotransmitter receptors on the postsynaptic surface. That's what we're going to talk about mainly in this tutorial. And so what we want to understand then are what are the consequences for activation of a receptor for a neurotransmitter on a postsynaptic cell. All right, so that's the challenge for the day. And I'll just remind you again that we have two classes of neurotransmitters, our small molecule neurotransmitters and then our peptide neurotransmitters. And these classes of neurotransmitters interact with two broad classes of receptors. So we will talk in some length about each class. We will begin with talking about our ionotropic receptors, which are receptors for neurotransmitters that are part of an ion channel complex. Hence we call these ligand-gated ion channels. And there are metabotropic receptors, which are receptors for neurotransmitters that are not directly part of ion channels, but rather these receptors trigger a set of metabolic activities that then might ultimately result in the opening or closing of separate ion channel molecules, as well as a variety of other postsynaptic activities. Our small molecule neurotransmitters typically interact with both ionotropic and metabotropic receptors, although that may not be universally true of all small molecule transmitters. Certainly our major important neurotransmitters like glutamate and GABA interact with receptors from both categories, whereas our peptide neurotransmitters interact with metabotropic receptors. Okay, so let's begin to consider what exactly these receptors are and consider their structure and functional properties. So let's begin by considering the ligand-gated ion channel. So as I said, the essential relationship here to understand is that the actual receptor sites themselves that interact with the neurotransmitters are part of an ion channel complex. Okay, so the receptor is part of the same molecular structure that forms the ion channel. And because of this, these are the receptor interactions that mediate the most rapid effects on postsynaptic currents. So as we go, I'll remind you that in order to understand the effect of any given neurotransmitter substance, whatever it may be, what we really need to know is something about the receptor to which that neurotransmitter binds. And we need to know something about what other receptors might also be present in the same postsynaptic cell in order to understand the physiology of that transmitter molecule. So we'll see examples of that as we go along here in this tutorial. All right, so let's consider the general molecular structure of ligand-gated ion channels, and we'll use the ligand-gated ion channel that we know best as an example. That would be the nicotinic acetylcholine receptor. So this is the receptor for the endogenous neurotransmitter acetylcholine that is also occupied and activated by nicotine. So this structure has a characteristic set of subunits that come together to comprise this ionotropic receptor. Typically, there are 5 subunits that come together to form the functional nicotinic acetylcholine receptor. Each subunit has an N-terminus domain to it that binds the actual neurotransmitter acetylcholine. So the N-terminus is out here somewhere in the extracellular space. Attached to that N-terminus is some number of membrane-spanning helical regions, and then there is some kind of cytoplasmic extension that often is a site where the receptor subunits can interact with metabolites and other kinds of molecules within the cytoplasm of the cell. So that's just one subunit. A functional channel requires the aggregation of multiple subunits, typically 4 or 5. Here in this example, we see 5 subunits that have aggregated. 2 alpha subunits, a beta subunit, a delta, and a gamma subunit. And they all differ slightly in their molecular structures. But together they aggregate to form a central pore that contains the filters that result in the selectivity of this ion channel. Now, as a class, ionotropic receptors are not as selective as our voltage-gated ion channels are, so as we'll see, multiple ions can pass through the pore of this channel. Now, what makes this a ligand-gated ion channel is the fact that channel opening is gated by the binding of the neurotransmitter to its receptor. So this ion channel is not particularly voltage sensitive. It doesn't have a voltage sensor motif among its membrane spanning domains. Rather, the gating mechanism is modified by the interaction of the transmitter with the receptor. That induces a conformational change that opens the pore of the channel, allowing for the passage of ions. Now, there are a variety of important examples of ionotropic receptors that we'll be talking about throughout the course. I've just been talking about the nicotinic acetylcholine receptor. There's a family of receptors for glutamate that are in the ionotropic category. There's a receptor named AMPA, AMPA, for the pharmacological agent that best activates that receptor. Likewise, there is a receptor called the NMDA receptor, N-methyl-D-aspartate receptor, and another one called the kainate receptor, all named for the exogenous ligands. Of course, the endogenous ligand for all 3 is glutamate. There's the receptor for the principal inhibitory neurotransmitter of the brain called GABA, gamma-aminobutyric acid. There are actually a variety of GABA receptors. Here are shown some of the subunits that come together to form what's called the GABA-A receptor channel complex, and so on. Receptors for glycine, for serotonin, and then for the purinergic transmitters such as adenosine. Now, what you'll notice from this chart here is that there are multiple subunits that can come together to form an ion channel, and combinations of subunits can form channels that have slightly different kinetic properties with respect to channel opening and closing, or perhaps different kinds of modulatory sites that are presented to the cytoplasmic surface, or perhaps different affinities for the endogenous ligands. So one can have different kinds of functional properties result from the aggregation of different kinds of subunits that come together to form the entire receptor channel complex. Let's look a bit more closely at one important ionotropic receptor, and that is the receptor for the inhibitory neurotransmitter GABA. So what we're looking at here is the typical GABA-A receptor channel, and it's comprised of 5 subunits that come together. In this case, an alpha, 2 beta subunits, a gamma, perhaps a delta or another alpha that have come together here. And like the nicotinic acetylcholine receptor, these five subunits aggregate to form a pore through which ions may pass. But what I want you to notice here is really the complexity of this ion channel. There's a variety of binding sites for other substances that can modulate the efficiency at which these channels open or close. For example, there is a site for the binding of steroids, for the binding of barbiturate drugs. Many of our pills that we take at bedtime that can modify the way people fall asleep are acting at a site deep within this receptor complex here. There are sites on the extracellular side that likewise can modulate the flux of current through these channels. Again, drugs that are useful pharmacologically and are important drugs in the medical armamentarium. And a variety of other binding sites for drugs that are not illustrated here, including alcohol. Now, this particular ionotropic receptor allows for the passage of chloride ions. So chloride ions pass through a selectivity filter here, which are favorable for this monovalent anion to pass down its concentration gradient. As illustrated here, that concentration gradient is from inside the cell to outside the cell, which is typical of an immature neuron, as we'll talk about in a little while. Now, let's talk about the currents that pass through ligand-gated ion channels. And in order to understand the principles involved, we need to revisit Ohm's law. So you will recall that we talked about Ohm's law as being V equals IR, and we were able to rearrange that in terms of current and indicated that current equals the inverse of resistance, which is a conductance term, times V. And that V term we could expand in terms of the driving force for a permeant ion as the difference between the membrane potential and the reversal potential for the permeant ion. So we can apply the very same principles to the movement of ions through a ligand-gated ion channel. What's different here is that the channel is gated not by voltage, but by the binding of a neurotransmitter to its receptor. So when we do that, what we see is that the current, which here is called PSC for postsynaptic current, is equal to the ligand-gated conductance, the G term, times the driving force. Now, if the conductance of this channel happens to be greater than 0, this is what would happen when a neurotransmitter binds, then we would expect there to be current provided that we have a driving force. If there's no driving force, that is, if the membrane potential is precisely the reversal potential for the active conductance, then even though we may have a significant value of conductance, then there will be no postsynaptic current if the driving force term equals 0. Okay, now I want us to consider how this version of Ohm's law applies to understanding the conductance of the nicotinic acetylcholine receptor that we have at our end plates on our muscle fibers. So let's turn our attention to the neuromuscular junction. So this is the place where our alpha motor neurons synapse upon our skeletal muscle fiber, as illustrated over to the right. And what we see here is an axon that makes contact with a muscle fiber. And we can experiment upon this preparation. Now, I'd like to take you through some data that are derived from such experiments. So notice that we can stimulate the axon with a microelectrode. We can voltage clamp the muscle fiber with a voltage clamp amplifier. So we can measure the membrane potential, we can pass current to establish whatever membrane potential is of interest to the experimenter. And so what we're looking at here is a recording of end plate current. So this is a kind of a postsynaptic current recorded from a muscle fiber. And notice that the muscle fiber is voltage clamped at different levels, from a rather hyperpolarized potential of 110 millivolts all the way up through an extreme depolarized potential of plus 70 millivolts. And when we first look at the extreme hyperpolarization, what we find is that there is a significant inward current. So again, negative current here by convention is an inward current carried by a cation. So with 110 millivolts of hyperpolarization, stimulating this axon leads to a strong inward current. If we cut that hyperpolarization in roughly half and now set the membrane potential to minus 60 millivolts, we have about half as much inward current flowing into that muscle fiber. Now notice what happens when we voltage clamp to 0 millivolts. We see essentially no current at all in this muscle fiber when we stimulate the axon. So keep that in mind. And finally, if we are to depolarize the muscle fiber to plus 70 millivolts and then stimulate the axon, what we see is not an inward current at all, but an outward current. So this behavior should look somewhat familiar to you because we saw similar behavior when we considered the impact of membrane potential on the currents that pass through voltage-gated ion channels. Only here, the ion channel is gated not by voltage, but by the binding of acetylcholine to its receptor. So note, for example, that we have inward currents at hyperpolarized potential, outward currents at depolarized potential, and no net current at 0 millivolts. So that should perhaps give you some insight as to what might be the currents that are flowing through this ion channel. Now, in order to help you think through that, I want you to be able to relate our version of Ohm's law that we're revisiting here in the context of looking at end plate currents at the neuromuscular junction. And I want you to think about the following set of study questions that you're about to see, and they're also prepared for you at the end of the tutorial notes. So why don't you take a few minutes now and pause the recording and consider the following set of study questions. So how did you do? Did that make sense? Did you have to back up a few minutes in the tutorial and look back at the data that we just went through? Well, if you did, then that's great. But if you found it a bit confusing, then hang with me now. Let's look at a similar experiment, only now taking it to the level of the passage of current through a single ion channel. Okay, so let's see what happens when we focus again on the nicotinic acetylcholine receptor and the movement of ions through that receptor channel. So what we're going to do is to consider the flow of current that can be recorded while this membrane is clamped at different membrane potentials. So let's begin at minus 100 millivolts. So what can be recorded is a strong inward current that results in a depolarization of the muscle fiber. So we're still at the end plate, so we're going to call this an EPP for an end plate potential. But it's just the membrane potential of the muscle fiber. Okay, and it's depolarizing relative to rest. So why is it depolarizing? What explains the strong inward current? Well, sodium ions are going to pass through this ion channel. This channel, because of its molecular properties, is permeable to both sodium and potassium, but at minus 100 millivolts, there's a huge driving force for sodium, but almost no driving force at all for potassium, because 100 millivolts is very near the Nernst equilibrium potential for potassium. So making the inside of the cell that negative is going to draw in the positively charged sodium ions, which are going to want to flow into the cell anyway because of its concentration gradient. So if we have slightly less polarization, minus 90 millivolts, maybe now we are just a little bit less polarized than the Nernst equilibrium potential for potassium, what we'll find is still strong sodium influx. So we still record a net inward current and significant depolarization, significant end plate potential. But notice now that we also might observe a small efflux of potassium, much less than the much stronger influx of sodium, which is why we're still recording an inward current, but it's not quite the same magnitude that we saw at minus 100 millivolts. So now let's clamp this membrane at 0 millivolts. And at 0 millivolts, what we observe is no end plate current. And with no net end plate current, we have no end plate potential. Now that doesn't mean that there's no movement of ions. In fact, sodium is still being driven into the cell. We are nowhere near the Nernst equilibrium potential for sodium within the cell, so sodium is still going to be drawn in because of the electrical gradient and also because of its concentration gradient. In a similar fashion, but in the opposite direction, potassium is going to flow out of the cell. Potassium will be repelled by this depolarization of 0 millivolts, and it's going to flow down its concentration gradient. So there are ionic fluxes, but the key is that there is no net ionic flux. So the net current is 0. And in essence, at 0 millivolts, the influx of sodium ions is exactly counterbalanced by the efflux of potassium ions. That's why, in fact, there is no end plate current recorded. Okay, and lastly, let's depolarize the cell to plus 70 millivolts. So for muscle fiber, plus 70 millivolts is getting very close to the Nernst equilibrium potential for sodium, given the concentration of sodium inside and outside of a muscle fiber. So at plus 70 millivolts, the driving force for sodium is essentially nil. Because we are very close to that Nernst equilibrium potential, even though the conductance of the channel is wide open. However, we are far from the Nernst equilibrium potential for potassium, so there's a huge driving force for potassium. That's going to allow potassium to rush out of the cell. It's diffusing down its concentration gradient, but it's also being repelled by the positivity that we have imposed upon the inside of this muscle fiber. So, as a consequence, what we can record then is an outward current, and that efflux of positive charge leads to a hyperpolarizing end plate potential. Now let's return to the brain, and let's consider the ionotropic receptors that are found at glutamatergic synapses. Now, as we've already seen, there are at least 3 types of glutamate receptors. We'll focus primarily on 2 of them. The receptor for AMPA or AMPA, and the receptor for NMDA. When AMPA is placed on a typical neuron in the brain, what we see is a very quick current that flows into the cell, typically leading to an excitatory postsynaptic potential. So we call this an excitatory postsynaptic current. So very sharp, very large inward current that happens very rapidly when AMPAs are applied to a neuron. If the compound N-methyl D-aspartate is applied, what we find is also an inward current, but it's not as large in amplitude, and it's a little bit more slowly developing, and it lasts a bit longer than the very fast, sharp AMPA current. So these different profiles reflect the activation of different types of receptors, both of which bind the endogenous ligand glutamate. So let's look in a bit more detail now at the behavior of one very important ionotropic receptor for glutamate, and that's the NMDA receptor for glutamate. So here in the upper right, we see an illustration of this receptor. It is a receptor that is formed by the aggregation of typically 4 subunits. It has a binding site for glutamate on some of the subunits, and sites that bind other types of molecules. I mentioned that glycine is a co-transporter in the central nervous system. There's a binding site on these subunits for glycine. So in order for the channel pore to open, both glutamate and glycine have to interact with their receptors on this channel complex. But there are other binding sites as well. In particular, there is a site deep within the pore of this channel where magnesium ions can aggregate. And this is a problem for the conductance of the channel, because even if the pore of the channel is opened by the binding of glutamate with the coagonist glycine, current is not going to flow through this channel if magnesium is present. Magnesium essentially blocks the flow of current through the channel. However, it's possible to get rid of that magnesium, and the way that happens is by depolarizing the membrane. So if the inside of the cell becomes more positive, that positivity can expel this magnesium ion out from the pore of the channel. When that happens, now the cations that can pass freely through the selectivity filter of the channel are free to do so. And those include sodium and calcium ions that can enter the cell, and potassium ions that can leave the cell. So this is a ion channel that's permeable to cations, but not particularly selective for one kind of cation or another. And importantly, the conductance through this channel is influenced by magnesium ions. When magnesium is bound, there's no conductance. When magnesium is expelled, then we see conductance. This relationship is charted below in the graph that relates the excitatory postsynaptic current to the potential of the postsynaptic membrane. And what we see is that when there is the presence of the transmitter glutamate and magnesium ions, there is little to no excitatory postsynaptic current recorded at hyperpolarizing potentials. But as the membrane potential begins to depolarize, when we get into this range here, now we begin to see some excitatory postsynaptic current passes. Essentially what happens is the magnesium block is relieved, and positive cations, sodium and calcium, can enter the cell. But notice that as we begin to depolarize towards 0 millivolts, the current reverses. Now, look at the behavior of the excitatory postsynaptic current if we remove magnesium from the solutions bathing the cell. In that situation, we have a relatively linear relationship between current and membrane potential, which is what one might expect if there were not this blockade effect due to the binding of magnesium ions to the inside of the pore of the ion channel. Now, before we leave our NMDA receptor channels for the present time, I just want to highlight the fact that the ion channel allows for the permeation of calcium. Now, this may not seem very important at this juncture of the course, but it is soon to be extremely important because, as we'll see in a couple of tutorials, calcium is critical for mediating synaptic plasticity. And the influx of calcium, whether we have a rapid, significant rise in intracellular calcium, or perhaps a slower and less significant rise in intracellular calcium, could make all the difference in the world between strengthening a synaptic connection and weakening that synaptic connection. And as we now understand, the NMDA receptor channel is a key coincidence detector of postsynaptic depolarization and presynaptic activity, which is critically important for understanding how synapses increase or decrease their synaptic strengths. So we'll come back to the NMDA receptor when we talk about synaptic plasticity and specifically focusing in on the important role of calcium. Okay, well, we're ready to shift gears now and talk about our metabotropic receptors. So again, these are receptors for neurotransmitters that are not ion channels. Rather, these receptors interact with a series of second messenger pathways within cells that lead to a variety of postsynaptic effects. Perhaps the opening and closing of ion channels, but maybe a great diversity of other kinds of postsynaptic effects as well. So here's a typical example of a metabotropic receptor, also known as a G protein coupled receptor, because the receptor itself is a monomeric integral membrane protein that is associated with G proteins on its cytoplasmic surface. When the receptor is activated by the neurotransmitter, these G protein systems then become active. And those G proteins then can interact with effector proteins, and those effector proteins can produce a variety of intracellular messengers that potentially can gate some ion channel. So in this example, an ion channel does indeed open following the binding of a neurotransmitter. So between the binding of the transmitter to a receptor and the opening and closing of an ion channel in this example are a series of metabolic reactions, and that takes time. So the metabolic steps are what accounts for the slower action of metabotropic receptors on postsynaptic conductances relative to our ionotropic receptors, where the effects are essentially instantaneous with the binding of a neurotransmitter to its receptor. So let's look at a couple of different examples. So the receptor itself is typically a monomeric integral membrane protein, but the intracellular second messenger systems related to G proteins can be quite diverse. We have our more canonical heterotrimeric G protein systems illustrated to the left, and so activation of G proteins alpha as well as the beta and gamma G proteins can go on and mediate a variety of postsynaptic effects interacting with effector proteins, some of which may produce second messengers themselves, others which may activate enzymes that can lead to the phosphorylation of target proteins and the activation of a variety of systems within postsynaptic cells. Other receptors for neurotransmitters are associated with monomeric G protein systems, such as the RAS system. And activated RAS can go on and mediate a variety of postsynaptic effects itself. Here's some specific examples. Are receptors for many of the biogenic amines, such as dopamine, norepinephrine, serotonin. They interact with G protein coupled receptors. Illustrated to the left is the beta adrenergic receptor for norepinephrine. It activates a GS version of the heterotrimeric G protein system, which leads to the activation of adenylyl cyclase and an increase in the amount of cyclic AMP that's produced within the cytoplasm. Cyclic AMP is an important second messenger system that can then lead to the increased phosphorylation of target proteins. Glutamate binds not only to the ionotropic receptors like the AMPA and NMDA receptor, but also to metabotropic receptors. Here, glutamate is binding to a receptor that activates GQ proteins, and these can lead to the activation of phospholipase C and the activation of other kinds of second messenger systems, such as the IP3 pathway and the diacylglycerol pathway, again leading to the phosphorylation or perhaps the dephosphorylation of target proteins within the cell. And then lastly, we see that dopamine binds to metabotropic receptors. We'll talk more about this when we talk about the circuitry of the basal ganglia. Dopamine is binding to a D2 receptor here, which interacts with a GI protein, in this case leading to the inhibition of adenylyl cyclase and a reduction in the levels of cyclic AMP. and that leads to a decrease in protein phosphorylation in the target site. Now, one benefit of the G protein-coupled receptor family is that there can be tremendous amplification of the signal as these metabolic steps play out. So imagine activation of a single receptor leads to the production of a fairly large number of G proteins. So there's amplification in this step. Well, in the pathway, for example, following beta-adrenergic receptor activation, these G proteins can activate adenylyl cyclase enzymes, which then produce cyclic AMP. So another significant amplification step happens where a large number of cyclic AMP molecules are derived from the activation of a smaller subset of adenylyl cyclase molecules. These cyclic AMP molecules then can interact with protein kinases, which leads to the massive phosphorylation of numerous target proteins. So significant amplification is the end result of activating a G protein-coupled receptor. G protein-coupled receptors can not only activate conductances in the postsynaptic cell, they can also interact with transcription factors that can modify gene expression. And these kinds of interactions mediate the longest lasting effects of G protein-coupled receptor activation. So there are pathways that follow the activation of heterotrimeric G protein systems that lead to the activation of regulatory elements such as the CREB binding domain in our genes. And activation of CREB can lead to modification of gene transcription. Similar CREB modifying pathways can converge upon the same machinery within the nucleus through different pathways. Here's an example of a RAS-mediated pathway also activating CREB. Sudden rises in intracellular calcium likewise can interact with calmodulin-dependent kinases to activate CREB. So there may be a variety of ways that we can influence the transcription of the genome based on the interactions that are happening out at the plasma membrane of a neuron. And G protein-coupled receptors are one important means for doing so. If we look at the molecular structure of G protein-coupled receptors, what we find are typically 7 transmembrane domains. These 7 transmembrane domains are characteristic of this gene family, and so we now know that they encode a variety of G protein-coupled receptors that show up in some surprising places. So what they have in common is that they are a monomeric protein. There is an extracellular domain, which typically includes an N-terminus and a site for the binding of a neurotransmitter. In this example, what we're seeing is the muscarinic variety of acetylcholine receptor. There is, of course, a cytoplasmic side of the molecule that interacts with the G proteins. So we expect to see that as well. And again, the defining feature are these 7 transmembrane domains that run through the plasma membrane. So, as I said, there are a variety of G proteins, and they show up in some surprising places. We'll find G proteins, for example, involved in sensory transduction in some of our special sensory systems, including the olfactory system and even the visual system. So the transduction of photons into electrical energy requires the activation of a G protein-coupled receptor system. So there's a variety of metabotropic receptors for a large family of small molecule neurotransmitters, as well as for our peptide neurotransmitters. And the complexity of this system is just now beginning to be appreciated. What's important for you to take away is the idea that the metabotropic receptors give rise to these more slowly evolving but potentially much more long-lasting effects of the binding of a transmitter to its receptor. So now that we've talked about these diversity of receptors that are present that interact with these neurotransmitters, I think you can understand why we cannot judge the physiological effect of a neurotransmitter by only knowing something about the identity of the transmitter and the kind of chemical structure of that transmitter. No, that's not enough. We have to know about the receptor in order to understand the physiological effect. And that receptor might be an inotropic receptor in mediating a fast effect on a postsynaptic conductance, or it might be a metabotropic receptor in mediating a slowly evolving effect that impacts a postsynaptic conductance but could also alter gene transcription and thereby have a long-lasting effect on that postsynaptic cell. Well, we're going to come back to some of these themes, and we will return to our metabotropic receptor family. As I mentioned, they show up in some pretty surprising places. We'll discover G-protein coupled receptor systems in our special sensory pathways, such as our olfactory system and in our visual system. And perhaps you'll be surprised to discover that the act of seeing requires the activation of a G-protein coupled receptor, very much like the same kinds of receptor structures we've just been looking at. So stay tuned for that. But meanwhile, now that we understand some of the basic molecular and cellular mechanisms that are responsible for chemical neurotransmission, we're ready to talk about the integration of electrical signals in a postsynaptic cell, and then that will allow us to talk more about synaptic plasticity and how the effectiveness of a synapse can be altered by its very use. So I look forward to having those discussions with you in the next few tutorials. Hi everyone, welcome again to my home. It's a nice cool evening and really a lovely day here in Durham, North Carolina today. Maybe you can tell since my last tutorial I had a haircut, so I'm especially feeling the nice cool weather this evening, and I'm happy to spend a little bit of time with you talking about synaptic integration. So we continue to talk about our important core concept in the field of neuroscience that pertains to the communication that happens among neurons and between neurons and peripheral structures via the generation of chemical and electrical signals. Specifically, our learning objective today is to discuss the concepts of excitatory postsynaptic potentials and inhibitory postsynaptic potentials. And what I want you to be able to do is to be able to define these terms in terms of the reversal potential for the postsynaptic current and the threshold for generating an action potential. Now, I trust all of that will be clear as we go through the tutorial together. And secondly, my objective is for you to be able to describe how postsynaptic potentials can summate within a neuron, both in space and in time. All right, well, in order to address this shorter tutorial on this subject tonight, I want us to start by reviewing the mechanisms of chemical synaptic transmission. Now, you've had a few tutorials on this subject, so I think things ought to be fairly clear, so I'll go rather quickly through this. As you will recall, an action potential will invade the end of an axon where we find our presynaptic terminal, and a wave of depolarization then conveys positive charge along the length of this presynaptic terminal. And once that positivity reaches our voltage-gated calcium channels, they will open and calcium can rush into that presynaptic terminal. And calcium in the presynaptic terminal is the key trigger that leads to the fusion of docked vesicles, allowing their neurotransmitter to then passively diffuse out into the synaptic cleft. And I'll just remind you that this happens when calcium interacts with synaptotagmin, and that leads to a twisting and pulling together of that snare complex that results in the fusion of that vesicle with the presynaptic terminal membrane. And that fusion event creates a pore so that the transmitter can diffuse out. And then once we have a transmitter in the synaptic cleft, then that transmitter can interact with receptors. In this illustration, we have ionotropic receptors. And that's really going to be our consideration this evening. You will recall from past tutorials that there are also metabotropic receptors for transmitters that mediate longer lasting effects. And surely that's part of what we have in mind when we talk about the integration of postsynaptic signals. But for this tutorial, I want to focus on the integration of currents that are mediated via the activities of ligand-gated ion channels. And I want us to think about those currents that are more likely to lead to the generation of an action potential in this postsynaptic neuron as this wave of depolarization reaches that postsynaptic terminal. Or I want us to think about those conductances that might actually make it less likely for that postsynaptic action potential to be generated. Okay? So on the one hand, we're going to talk about action of neurotransmitters that elicits what we call an excitatory postsynaptic potential. And on the other, we're going to talk about activities of neurotransmitters that give rise to what we call an inhibitory postsynaptic potential. So forgive me if I use that jargon quite a bit today, EPSP and IPSP. This is jargon that we will find throughout the literature in the world of neuroscience, especially as it pertains to neurophysiology. So when you hear that EPSP, IPSP, think of excitatory postsynaptic potentials and inhibitory postsynaptic potentials. So I want us to spend just a little bit of time with what I consider to be a very important figure from the textbook that we've been reading along with this course so far. It's figure 5.21, and it pertains to these concepts of excitatory postsynaptic potential, inhibitory postsynaptic potential, and how we can understand and even define those terms relative to the reversal potential. Remember that concept? That comes way back when we first introduced the Nernst equation and talked about the equilibrium potential for a permeant ion. Well, we're going to bring back that concept and now apply it to the conductance that passes through a ligand-gated ion channel. So what we have illustrated in part A of this figure is an excitatory postsynaptic potential. So we might think of this as a postsynaptic potential that is secondary to the release of, let's say, glutamate from a synapse, or maybe a collection of synapses that are converging upon the same dendrite of a postsynaptic neuron. Well, as glutamate binds to its ionotropic receptors, for example, AMPA receptors, those receptors open and sodium rushes into the cell. And as a consequence, what we see is a depolarizing event. We call that depolarizing event an excitatory postsynaptic potential. So why do we call it that? Well, here's the definition. The reason why we call it an excitatory postsynaptic potential is that the reversal potential of the active conductance is well above the threshold for firing an action potential. Now, a couple of tutorials ago, we looked closely at the conductances that pass through the nicotinic acetylcholine receptor, and because both sodium and potassium can pass through that receptor, the reversal potential for the net current flow, that is the net conductance, was around 0 millivolts. And that's very much the same situation that we have here for this conductance that is gated by glutamate. So the reversal potential is about 0 millivolts, and that's because sodium is entering the cell through the AMPA ionotropic receptor, and potassium is leaving the cell. So the voltage, that is the membrane potential, at which there's no net current flow, is 0 millivolts. Okay, so what does that mean for this particular synapse that has this effect on this neuron? Well, what this means is that if this synapse, if this input to this cell in question from which we are recording if this synapse could have its way with that neuron, and pardon me for that language, but I think it helps, if that synapse can have its way with that postsynaptic neuron, that postsynaptic neuron will be depolarized to the reversal potential of that active conductance. Now, the synapse doesn't have its way because it's, like most synapses in the brain, a weak synapse. Maybe there's not a lot of glutamate that's being released. Maybe there aren't a lot of AMPA receptors to receive that glutamate. However, the reversal potential for the AMPA-mediated conductance is near 0 millivolts. So that means that if this particular synapse could completely capture that postsynaptic neuron, it would depolarize it to the reversal potential. Well, as I just mentioned, most synapses are not that powerful. They're actually rather weak, and they typically only allow for a postsynaptic membrane to depolarize by a few millivolts. And that's the story that we have here. If there is a single active glutamate synapse, even a very strong and powerful glutamate synapse, we may only see an excitatory postsynaptic potential of some modest value. But if we imagine the concurrent stimulation of a number of inputs, we might very well have a depolarization that hits that threshold for firing an action potential. So do you remember what threshold is? Threshold is when more sodium ions are rushing into the cell than potassium ions are leaving the cell. When we reach that threshold, then an action potential can fire. Okay, so let's put this together now. So we activate a glutamatergic synapse, or perhaps a collection of glutamatergic synapses on this cell. The cell begins to depolarize because sodium is rushing in to the cell through the AMPA receptor channel, and its reversal potential is around 0 millivolts, so we've got a large conductance because the channel's open, and we've got a large driving force, right? Because we're pretty far away from that reversal potential when we start at rest. Well, as this cell begins to depolarize, now we start opening up our voltage-gated sodium channels. And that's what leads to triggering this action potential that we record here. Okay, so the binding of glutamate to the AMPA receptor is what gets us started, and then the action potential is generated through the usual means, through voltage-gated sodium channels, accounting for the sharp rising phase, and then sodium channel inactivation begins the falling phase. That gets reinforced with the opening, the slower opening of the voltage-gated potassium channels. All right, that is a typical glutamate synapse within the brain. Now let's consider a different neurotransmitter, the neurotransmitter GABA, gamma-aminobutyric acid. Now, if you remember, GABA binds to a receptor that's part of an ionotropic receptor, and that receptor is permeable to chloride ions. And in most mature brain cells, there's more chloride outside than inside. So that means when the chloride channel is open, chloride ions are going to want to enter the cell. Okay? And if we are making the inside of the cell more negative because of the influx of chloride ions, we might expect some hyperpolarization. And that's exactly what we see here. If we activate a GABA synapse, there may be a small hyperpolarization. And the reason is that we are trying to drive that postsynaptic membrane to the reversal potential of the active conductance. Same principle that we talked about for the glutamatergic synapse, only now we're applying it to the situation of a GABA synapse. So notice where the reversal potential is for this particular neuron in question. It's actually hyperpolarized relative to rest. So when the conductance opens, the concentration gradient is such that it will hyperpolarize by a few millivolts this membrane potential. That's what we call an inhibitory postsynaptic potential. Not because it's hyperpolarizing, but because the reversal potential is below threshold. All right, just to reinforce that point, let's look at the third example. This also is a GABA synapse, only because of the concentration gradients inside and outside this particular cell, the reversal potential for the active conductance is actually in the depolarizing direction. But get this, it's still an inhibitory postsynaptic potential. Why? Because the reversal potential of the active conductance is below threshold. Okay? I hope you're catching on to this. So again, let me use that somewhat loose language of saying if that synapse with that particular conductance could have its way with the postsynaptic neuron, it would clamp the membrane potential of the postsynaptic neuron at the reversal potential. Okay? No more, no less, at the reversal potential. So, in theory, the most depolarized this neuron, shown in panel C, could ever possibly get with this active conductance is the reversal potential, which is below threshold. So what we see, in fact, is when we activate that synapse, a depolarizing postsynaptic potential, but it's still an inhibitory postsynaptic potential. So please don't make the association of depolarization with excitatory postsynaptic potential, because you'll be missing the concept entirely. I want you to associate the idea that the reversal potential of the active conductance is what defines a postsynaptic response as being an EPSP or an IPSP. So if the reversal potential of the active conductance is above threshold, then that is an excitatory postsynaptic potential. If the reversal potential of the active conductance is below threshold, then that is an inhibitory postsynaptic potential. Now, if you're following along with the tutorial handout that I've given you, I'm asking you a question at this point. I'm asking you, can the same neurotransmitter give rise to excitatory postsynaptic potentials and inhibitory postsynaptic potentials? Well, the answer is yes, and the reason has to do with the definitions that I've just given you, which are illustrated over here in panel D. So if the difference between an EPSP and an IPSP is the reversal potential, then you should now have some intuition that tells you that the reversal potential is really a function of the concentration gradient of the permeant ion. So suppose that concentration gradient were reversed. Well, we may take a reversal potential that is above threshold and now place it below threshold, or vice versa. Well, this is the case for GABA. GABA is usually described as the principal inhibitory neurotransmitter in the brain. But about 10 or 15 years ago, we realized that this might not be the case in the developing brain, and here's why. When we look at the pumps for establishing chloride gradients in the developing brain, we realize that there is actually a difference. There's a bit of a switch that takes place during brain development, at least for most neurons in the model species for which this has been studied so far. What we find is a particular type of chloride transporter that is expressed in immature neurons, and then as these neurons grow up a bit, a different transporter is expressed, and that transporter reverses the chloride gradient. So in an immature neuron, we typically have high chloride concentrations in the cytoplasm and low chloride concentrations in the extracellular spaces. But once this gene switch is turned on and a different type of transporter is now inserted into the membrane as the neurons develop in postnatal life, now we have low chloride concentrations within the cytoplasm and much higher chloride concentrations outside of the cell. And this has a very predictable impact on the reversal potential of the GABA A channel conductance. The reversal potential in the immature neuron is above firing threshold for most neurons. whereas following the reversal of that gradient, as neurons get older and more mature, we find that the reversal potential for this concentration gradient of chloride now falls below threshold. So in the immature neuron, GABA is an excitatory neurotransmitter mediating excitatory postsynaptic potentials. But in the mature neuron, GABA typically is going to mediate inhibitory postsynaptic potentials. Okay, and again, the key difference is the expression of different transporters that can change the gradient for the permeate ion. Here's some electrophysiological data that makes this point. In the left part of panel C is a recording from a 6-day-old neuron in a mouse. And what we find is that with application of GABA, there is a series of action potentials that rise upon this envelope of depolarization. This is an EPSP leading to the generation of action potentials. Now, if we were to wait a few days later, as these neurons mature and apply GABA, now we see that there is a hyperpolarization of the membrane. And the difference is a pump that reverses the gradient from high intracellular chloride levels to low intracellular chloride levels across the first few weeks of postnatal life, at least in the mouse model. We have every reason to believe that the same biology is happening in the human brain during development. We just don't yet know exactly when that shift in expression of chloride transporters takes place. All right, now I'd like to switch gears just a little bit now and talk about the integration of both excitatory postsynaptic potentials and inhibitory postsynaptic potentials. And in figure 5.22, we see a very simplified representation of what we find in the brain. We have a postsynaptic neuron that's receiving a number of inputs. We're illustrating 3 of them here. So we have an excitatory input that we call E1, another excitatory input that we call E2, and then an inhibitory input. Now, of course, if we're talking about a typical brain cell, there would be thousands of these inputs, some of which would be excitatory, some of which would be inhibitory. But we're going to keep things real simple and only talk about 3. So let's consider what would happen if either that first or the second excitatory input alone were to fire an action potential. Most likely what we would see is a subthreshold depolarization. So that's what we have here if we activate either of these 2 inputs. We have a small depolarization of a few millivolts. And this really is more typical of what we see in the brain. No one synapse is that strong. So in order to achieve firing threshold for the generation of an action potential, we need to have multiple excitatory inputs summate their postsynaptic potentials in the same region of the dendrite at the same time. This is what we call summation. So look at what happens if now we activate excitatory input 1 and 2 concurrently. Now we can generate a postsynaptic potential that sums up. And the sum of these 2 potentials is sufficient to achieve action potential generation threshold. And as a result, we have this nice action potential that fires. Okay, well, let's now think about our inhibitory input that we have on this neuron. So let's imagine this is an input releasing GABA, and let's imagine it's a mature neuron. So when the GABA A receptor channel opens, chloride is going to rush into the cell, and that very well could cause a hyperpolarization of that neuron. And in fact, that's exactly what we see here. When we activate input I, the inhibitory input, we see a small inhibitory postsynaptic potential. And again, it didn't matter if it was hyperpolarizing or depolarizing, as long as the reversal potential of the active conductance was below threshold. It could be here, or it could have been here, but as long as it's below the threshold, then it will be an inhibitory postsynaptic potential. Okay, well, now let's get a bit more realistic. Let's have both excitatory and inhibitory inputs firing more or less the same time. And what we see is that these inputs will sum algebraically. So if we add one excitatory, one inhibitory input together, then the result is going to be the summation of the two. And in this example anyway, we will see at best a subthreshold depolarization. That might still be the case if we were to add the two excitatory together with an inhibitory. So I think you get the idea here. That is that no one synapse is typically strong enough to result in the generation of an action potential in the postsynaptic neuron, but we need to have many synapses, perhaps even tens or hundreds or thousands of excitatory inputs converging on the same neuron at the same time in order to depolarize that membrane sufficiently to reach threshold. Because we can be sure that there's probably some inhibitory activity that's converging at the same time as well. So we have to overcome the impact of that inhibitory input, even while we summate our excitatory inputs to achieve threshold. I'm distracted to my left just for a moment. It sounds like a deer has just walked by. It's dark enough that I can't see him. Could be an opossum, but it sounds like a deer. Actually, I think it is an opossum. Well, this brings us to the end of this shorter tutorial, and I hope it helps you put these concepts of excitation and inhibition in context, and I hope it allows you to better understand the neurophysiology of real neurons in the brain. Okay, so when we talk next time, we will be exploring together the fascinating phenomena of synaptic plasticity. I can't wait to have that conversation with you all. Welcome to this tutorial. This is the first of 3 devoted to the fascinating topic of synaptic plasticity. In this tutorial, we're going to talk about 2 particular mechanisms, one called long-term potentiation, or LTP for short, and the other long-term depression, LTD. This tutorial will be followed by one that will frame up our understanding of plasticity as it occurs in real brains, one spike at a time. And then lastly, we'll talk about the famous Heb's postulate and how our understanding of synaptic plasticity is framed up in those terms. Well, this topic relates to several key concepts in the field of neuroscience, and they are enumerated below. Neuroscience core concepts 2, 4, and 8. We will continue to talk about how neurons communicate through both electrical and chemical signals, and the plasticity that we'll be speaking of in this tutorial relates to plasticity of chemical signaling. And the point of plasticity is that it provides us with a means for life experience to change the nervous system. So we're beginning to talk about some of the most fascinating topics in the field of neuroscience, those topics that pertain to understanding how the experiences of our lives are transduced into permanent change in the nervous system. All of this holds great promise for the future. Fundamental discoveries promote healthy living and the treatment of disease. These topics that we will be exploring in the next few sessions are really giving us a glimpse into the future of medical neuroscience. As principles of synaptic plasticity are better understood, so can rational intervention be developed that will harness this amazing plastic potential of the central nervous system for therapeutic advantage. And for those of you that are getting into the healthcare industries, these are exciting times, because as basic neuroscience progresses in this domain of understanding these principles, so will our capacity to apply these principles for therapeutic good for the patients that you'll come to serve. So I'm privileged to be a small part of your education that will usher in this new frontier in the world of medical neuroscience. where we will understand how to optimize our capacity to change the nervous system of someone who is dealing with impairment or disease or disability, or perhaps someone without those conditions who wants to optimize their performance in whatever domain of life is of concern to them. So that's what's possible over the course of your lifetime and your practice as we come to better understand the brain basis of synaptic change. So I do have some learning objectives for you. I want you to be able to characterize general cellular mechanisms for synaptic change. In particular, I want you to be able to discuss the mechanisms of long-term potentiation. And I want you to focus on two glutamate receptors, the AMPA or AMPA subtype of glutamate receptor and the NMDA glutamate receptor. And I want you to discuss the role of these receptors in the induction and maintenance of long-term potentiation. Likewise, I want you to characterize long-term depression. And I want you to discuss the molecular basis for long-term depression in two important regions of the central nervous system, the cerebral cortex and the cerebellar cortex. Well, you may not need any motivation at all when it comes to this topic of synaptic plasticity. But again, for those of you that are pursuing careers in healthcare, I think it is especially pertinent that you learn these lessons well. One motivating statement, at least for me, has come from one of my colleagues at Emory University, who's been a leading thinker and researcher in the translation of basic science discovery in the domain of synaptic plasticity to intervention with people that are dealing with neurological disability. What Taub and his colleagues have written some years ago in a really excellent review in Nature Reviews Neuroscience, he wrote that behavior, that is, the sensory and motor experiences of our bodies, can have a profound influence on the function and organization of the nervous system, and that this effect can be manipulated to therapeutic advantage in individuals with central nervous system injury. So, understanding the rules of plasticity will increase our capacity to help people that are dealing with central nervous system injury and disease, because we will better understand how to take the sensory and motor experiences of their own bodies and use that capacity to reshape the structure and function of their nervous systems. So let's begin this discussion by making sure we really know what we're talking about when we use this word plasticity. Plasticity simply means the capacity of the nervous system to change. And we can think about this concept from a number of different perspectives, and I'd like to suggest a few of them. I'd like to suggest the temporal frame of understanding plasticity. Plasticity can be short-term, over the course of milliseconds to hundreds of milliseconds to seconds to minutes. It can be long-term. It can last minutes to hours to days and to even a lifetime. Just think back for a moment to some of your earliest childhood memories. It's amazing, is it not, that that is even possible? That some formative event changed the function, maybe even the structure of your nervous system in such a way that those events can be recalled now years later, in my case, decades later. Truly a remarkable achievement for a nervous system to have this capacity to store a lifetime of change. Now, of course, that's not to say that the process of remembering or recalling does not itself induce a new round of plasticity. Indeed, it does. But nevertheless, the basic information that was laid down in the initial event that you and I may be recalling is still with us. It's still present in the functional architecture of our brains. Amazing. So our discussion today will begin to get at an explanation as to what may have changed and how. We can also consider plasticity in spatial terms. That is, where does plasticity occur? Well, plasticity occurs at synapses. That's what we will talk about today. Plasticity might also occur within the structure and function of the neuron itself. That is, it may simply not be a change at the synapse, but perhaps a change in different compartments within the neuron. And the same could be said of glial cells. And in particular, I'm thinking about astrocytes that are important in modulating the activities of neurons within gray matter. Astrocytes themselves are subject to plastic responses induced by changes in their surrounding neural elements. Plasticity can affect the structure and function of entire neural circuits and entire systems comprised of multiple neural circuits operating in series and in parallel. Imagine the accrued impact of synaptic plasticity and cellular plasticity throughout an entire neural system, and the change can be profound. That change can ultimately be reflected in the organization of functional representations that we have in the brain. Consider, for example, the map of the body that we have in our somatic sensory cortex. That map is constantly being updated as we change the use of our body, or perhaps as the body becomes injured. So plasticity is a means by which we can constantly update and refresh the learning and the representation that we store even across an entire cortical area. Well, with such change pervasive throughout our organization of neural systems and neural circuits, it's not surprising then that plasticity should be the basis of basically everything we do in our nervous system that is predicated upon learning and change. So plasticity is the basis of memory. It's the basis of acquiring the motor skill that you may have acquired in life, be it in athletics or perhaps performance arts. Maybe it's in a musical instrument. Whatever it may be involving the acquisition of motor skill, there's been some plastic change at multiple places in your nervous system that has made that skill possible. The same would apply to cognitive skills. Any domain of learning requires that there be some plasticity. Certainly the development of language or the acquisition of new languages throughout life. Mathematics, for example, the ability to acquire a new skill and then apply that knowledge in problem solving. That's another example of plasticity in action, as would be any of the dimensions of the performing arts, as I mentioned a moment ago. And finally, again, putting it back into the context of healthcare, adaptation and recovery from injury or disability requires plasticity within the central nervous system. So let's just review some of the basic cellular mechanisms of plasticity that seem to apply to multiple forms of plasticity. And we'll see how these are played out in different contexts. So plasticity usually begins when neural activity triggers the activation of postsynaptic second messenger systems. And those second messenger systems usually involve an alteration in the level of intracellular calcium in the postsynaptic neuron. So calcium will be a key index that is used by the postsynaptic neuron to know which way to go with its plastic response. So calcium within the postsynaptic neuron triggers the activity of second messenger systems that often converge on one of two categories of enzymes. One category is the protein kinase family of enzymes. These are enzymes that phosphorylate target proteins, and that's often a mean of turning on that target protein. Another category is phosphatases, and phosphatases are enzymes that dephosphorylate target proteins. So it's this alteration in protein phosphorylation that mediates the early stages of synaptic plasticity, where new proteins don't need to be produced, so we've not yet really altered the overall protein complement of the postsynaptic cell. We've just altered the function of the proteins that already exist. However, for the long haul, there must be long-lasting change in synaptic strength that's brought about by other mechanisms, those mechanisms that involve a change in gene transcription. Well, all of these steps are common to multiple forms of plasticity, and one key differentiator of one form or another is going to be the level of calcium that's present in the postsynaptic cell. So pay attention to where the level of calcium becomes critical for determining what type of plasticity is observed. Okay, so now we're ready to talk about one particular mechanism of plasticity called long-term potentiation. But before we can, I need to introduce you to a model system that was foundational for the studies that first brought to light these mechanisms. And that model system is the slice through the hippocampus from a rodent used as an animal model. So these experiments can be done because when an animal is humanely sacrificed and its brain rapidly removed, tissue slices can be obtained from that brain, and those slices can actually be kept alive for some period of time, perhaps as long as a day. And during that time, those slices can be probed with microelectrodes, and axons can be stimulated and the activities of cells can be recorded. And this is what was done from slices obtained from the hippocampus of rodent models. So the hippocampus in the rodent is in the posterior part of the forebrain. It is something of a C-shaped structure with part of it in the dorsal aspect of the forebrain and the rest of it wrapped around the medial margin of the lateral ventricle as it extends in the posterior part of the forebrain. Now in primates, of course, the hippocampus has rotated further down into an inferior and forward position. So in primates, the hippocampus would be found in a position of the medial temporal lobe. But of course, rodents don't exactly have a temporal lobe. They have corresponding cortical regions, but not a lobular shape per se. Well, be that as it may, it's possible to obtain sections through this hippocampus and keep those sections alive in a dish, if you will, and manipulate the activities of those cells while recording different populations. And this is all done with microelectrodes. So there's a particular circuit that you should be introduced to in order to make sense of the experiments that I'd like to tell you about. And it involves basically a 3-neuron pathway through the hippocampus. And it begins with inputs that arrive into a part of the hippocampus called the dentate gyrus. And these inputs are coming from the cortex just outside the hippocampus and something called the entorhinal cortex, part of the parahippocampal gyrus, that you may recall from viewing the surface of the human brain from below. And these inputs make synaptic connections on a type of cell called the granule cell. The granule cell then sends synaptic inputs to a large pyramidal cell that's found in a region called the CA3 region of the hippocampus. This large pyramidal cell sends axons to several different places. One of them is to a different sector of the hippocampus called the CA1 region. And it's this pathway from CA3 to CA1 that we're going to focus in on and use as our model system. This axon that goes from CA3 to CA1 is called the Schaefer collateral axon. And it's possible then to put microelectrodes into this system and stimulate some number of these axons that might be converging on a pyramidal cell in the CA1 region. And the experiments that I'd like to tell you about are as follows. A particular microelectrode that is positioned along this pathway is going to be stimulated. So over time, there may be a brief barrage of electrical pulses applied. This may last just a couple of seconds at a very high frequency. And this is called a tetanus. So this kind of tetanic stimulation is what will be used to stimulate a pathway and induce long-term potentiation. Meanwhile, a second microelectrode can be positioned to activate a different set of axons that is present and functional, but just not stimulated with a tetanus. Now, to measure the impact of the tetanus in the pathway that's stimulated, as well as the parallel pathway that wasn't tetanized, it's possible just to provide a test shock, maybe about once every 30 seconds, and then record from this postsynaptic cell the amplitude of the excitatory postsynaptic potential, and see what happens to that postsynaptic potential. Does it stay the same? Maybe it increases. Well, this is the basic experiment that was performed. And the results are shown here. So pathway 1 is the pathway that received the tetanic stimulation. So before the tetanus, a single electrical shock gave rise to a particular excitatory postsynaptic potential of a particular amplitude and a particular rate of rise. But then after this tetanus, look at what happened. The excitatory postsynaptic potential in the same pathway in response to a single shock is about doubled in amplitude. So this is long-term potentiation. The difference in the amplitude of the response of that synapse to the test shock between the pre-stimulus condition and the post-stimulus condition. Something about that brief tetanic stimulation caused some fundamental change in the strength of this synapse. So now it has twice the impact on the postsynaptic cell than it did previously. Now, that was for the pathway that was stimulated with the tetanus. Look at the parallel pathway that was not tetanized. It's present, it's active in response to a single shock stimulus. It gives rise to a excitatory postsynaptic potential, just like the neighboring pathways, only now, before and after the application of the tetanus to pathway 1, there's no change in pathway 2. No change, no LTP. Here's another way to look at these data. Plotted here are the responses in these two pathways to the single test shock applied about every 30 seconds or so for about 15 minutes before the tetanus to about an hour afterwards. And what we see in pathway 1 is about a 2 to 3-fold increase in the amplitude of the EPSP over time. Now, there is some decay of this potentiation over the course of this experiment, lasting about an hour, but nevertheless, after about 1 hour of time, there's still about a 2-fold increase in the amplitude of that EPSP, the excitatory postsynaptic potential, in the pathway that was tetanized. In the pathway that was not tetanized, there's essentially no change. Now, these initial experiments were done at a time when slices in a dish could be kept alive for about a day or so. But what one really wanted to know was whether this potentiation might be a mechanism of learning and memory over a much longer period of time. And that required in vivo experiments. And indeed, this has been done. And we now have a compelling body of data suggesting that long-term potentiation may indeed be the mechanism by which information is stored across a lifetime. Well, this is not a lifetime, but for a rodent it's still a very long period of time within its lifespan. Notice that following the application of a tetanic stimulation, there is a significant increase in the amplitude of the excitatory postsynaptic potential recorded in vivo out for about a year's time. So now we're beginning to gain confidence that long-term potentiation may be one means of storing information at the synaptic level across a lifetime. Now let's consider a couple of properties of long-term potentiation that help us to understand some of the principles at play here. One principle is called specificity, and the other is associativity. Well, the principle of specificity means that in order for a pathway to be potentiated, it must be active. So imagine again the scenario where we have two pathways. One pathway is active and it's stimulated, and that synaptic connection is going to be strengthened with the application of that high-frequency burst of activity. Meanwhile, another pathway, pathway 2, that was not active at the time that pathway 1 was active, its synapse is not going to be strengthened. So in order for there to be potentiation of a pathway, there must be activity at that synapse between the pre and the postsynaptic partner. Meanwhile, other parallel pathways that are not active will not be strengthened. Okay, now we have the principle of associativity, and this is extremely important. Now imagine we've got two pathways converging upon the same postsynaptic neuron. One pathway is already a strong pathway. That pathway may then be stimulated and it can strengthen even more. Now, the second pathway is weak. That is, it has only a very modest effect on the postsynaptic neuron compared to the first pathway. But if the second pathway, the weak one, is activated concurrently with the strong pathway, then the weak pathway can be strengthened. And this is the principle of associativity. One reason why we think this is so exciting is that this principle of long-term potentiation potentially provides a paradigm for understanding associative learning, where some reinforcing event, if paired with an event of a neutral nature, can now acquire the significance of the reinforcer. And when we think in those terms, we recognize the basic paradigm of this experiment. The pairing of a strong pathway with a weak pathway can lead to the potentiation of both. Now, unfortunately, I need to digress for just a moment and clear up an unfortunate error in this figure. This figure, unfortunately, has duplicated the text from these two blocks. That should not have been. In the right-hand side of this figure, this word not should be scratched off. So I'm going to do that digitally by just flying in a little patch. So now we don't see that word not anymore. So in associativity, the pathway that has weak but concurrent activity can be strengthened along with the already strong pathway that was stimulated. So, to summarize these principles, specificity means that only active synapses are strengthened, and associativity means that coactive synapses may be strengthened. Now, what I've been telling you about long-term potentiation so far has been well studied in the hippocampus, and it's also been well studied now in many different regions of the cerebral cortex and even in subcortical structures. While the details may differ somewhat from one type of gray matter to another, the basic principles of long-term potentiation seem to be well established and seem to be present in recognizable forms in all kinds of different parts of the central nervous system when they've been discovered. Now, many of these mechanisms of long-term potentiation are dependent upon two important different types of glutamate receptors. And so now let's turn our attention to the mechanisms that support this long-term potentiation. And to do so, let's focus on these two particular glutamate receptors, one called the AMPA receptor for AMPA, and the other the NMDA receptor. These two different kinds of receptors, they're both ionotropic receptors for glutamate, which means that the site for binding glutamate is part of an ion channel. And both of these ionotropic receptors, therefore, can mediate rapid conductances in that postsynaptic cell. Well, let's look at how they both contribute to the induction and the maintenance of long-term potentiation. Now, let's consider first a synapse that has not been stimulated with that high-frequency train, so its synapse is relatively weak. And its impact on the postsynaptic cell is fairly modest. So if this synapse is firing at a fairly low rate, we would expect the postsynaptic cell to remain near its resting membrane potential. There may be a small depolarization that could be recorded, but we wouldn't expect very much at all with low frequency stimulation. So what's happening there is that the action potential in the presynaptic neuron would be sufficient to induce the release of glutamate from the synaptic vesicles that are present there. The glutamate would bind to its receptors on the AMPA and NMDA receptor channels. The binding of glutamate to AMPA would open up the pore, allowing sodium ions to come in. But again, with low levels of activity, we wouldn't expect a whole lot of depolarization to happen at this particular synaptic junction. Well, that would be a bit of a problem for our NMDA receptors, for, as you will recall, in the pore for the NMDA receptor channel, there's a binding site for magnesium ions. And that magnesium is going to block the conductance of that channel. Even though glutamate's bound and the pore is in its open configuration, the presence of magnesium is going to block the influx of ions. Well, that's what happens when the postsynaptic cell is near resting membrane potentials. However, should there be a strong depolarization of that postsynaptic cell, which would be expected if there would be a high-frequency train of activity at this synapse, then what we would find is a flood of postsynaptic glutamate out of that presynaptic terminal and perhaps out of surrounding synapses from the same input that might reach this postsynaptic dendrite, and that might lead to much more significant depolarization in this postsynaptic site. Well, as that happens, sodium's coming in through the AMPA receptors, perhaps a wave of depolarization is passively propagating along the dendrite, impacting the spine. There may be sufficient depolarization to expel magnesium from the pore of the NMDA receptor channel. Now, as you know, magnesium is a divalent cation, so if the inside of the cell becomes positive, then magnesium is going to be repelled out of that channel pore. That will allow that channel now to be available for the influx of sodium ions and calcium ions, as well as the efflux of potassium ions. If you remember what we learned a few tutorials ago about the NMDA receptor channel, you'll know that it's cation selective, but not particularly selective for which cations that flow. So all three of those cations are going to be involved. The one that I would highlight, however, is calcium. So calcium, very important molecule for reasons that will be clear in just a moment, enters through the NMDA receptor channel. And with a significant depolarization, then we would expect there to be a rapid elevation of postsynaptic calcium levels to a significant degree in this stimulated synaptic junction. That's important because the rapid elevation of postsynaptic calcium leads to the induction of long-term potentiation. Well, let's look at what we know about how that might occur. So Again, we're looking at a synapse that has been subject to a strong depolarization, perhaps via that high-frequency stimulation that may have been applied in an experiment, or some other means of depolarizing this postsynaptic spine. And under those conditions, what we'll see is a rise in intracellular calcium. And that rise will be sudden, it will be significant, and that intracellular calcium then becomes the key trigger of second messenger systems that result in the induction and the maintenance of long-term potentiation. So, intracellular calcium can interact with second messenger systems such as the protein kinase C system or the calcium calmodulin kinase 2 system, both of which will phosphorylate target proteins. And among the targets are the mechanisms that are involved with the trafficking of AMPA receptors from pools that are associated with vesicles within the postsynaptic spine to the terminal membrane. So as a consequence of this rapid increase in postsynaptic calcium, now AMPA receptors can be inserted in the postsynaptic membrane. And the more AMPA receptors we have in the postsynaptic membrane, the stronger is the response of that postsynaptic neuron to synaptic glutamate. This is a molecular picture of what long-term potentiation actually means at a postsynaptic spine. Well, one consequence of long-term potentiation, especially in the developing brain, is the awakening of silent synapses. Well, what is a silent synapse? A silent synapse is a synapse that is morphologically present, and it might be capable of function, except that it would lack AMPA receptors. And so here's evidence for one such silent synapse. Imagine stimulating an input to a postsynaptic cell while that cell is near its resting membrane potential, and there may not be any measurable postsynaptic potential recorded. So one might imagine that, well, maybe the site of stimulation doesn't connect to that cell. However, if the cell is depolarized with a voltage clamp, one might then record a significant excitatory postsynaptic current. And that is evidence that indeed there is a synaptic connection there. The required depolarization of that postsynaptic neuron implicates a synapse that is populated by NMDA receptors and no AMPA receptors. So what we might find, and indeed frequently we do find, especially in the developing brain, are these silent synapses where we have NMDA receptors present only with insufficient numbers of AMPA receptors to make this functional with low frequency stimulation. However, with the presence of stimuli that can induce long-term potentiation, and these kinds of stimulation events can accrue during the course of development, we see the insertion of AMPA receptors to accompany the NMDA receptors. And so now a silent synapse has been awakened into a functional synapse, which involves both the presence of NMDA receptors and now a complement of AMPA receptors sufficient to mediate the effect of synaptic glutamate, even without significant depolarization of the postsynaptic process. Here's another picture of what this might look like. Before long-term potentiation, stimulation of an input might lead to very little postsynaptic current, but after long-term potentiation, the same stimulus might lead to a significant inward current recorded at that synaptic junction. And this suggests that there has been the insertion of new AMPA receptors, possibly even the generation of a new synaptic connection between the presynaptic element and the postsynaptic dendrite. One can record from circuits in which there's a significant increase in the response of the postsynaptic neuron to glutamate. Again, indicative of plasticity, either the awakening of silent synapses or in some cases the construction of new dendritic spines in response to the growth of new synaptic connections. Well, for that to happen, there needs to be a mechanism by which synaptic plasticity over the short run can influence long-term change. And that requires not just the activation of second messenger systems that are already present within the postsynaptic spine, but the activation of systems that can trigger a change in gene expression. And that's possible following long-term potentiation. So in the early phase of long-term potentiation, we may see a calcium-dependent increase in the number of AMPA receptors that are inserted, as we've been discussing. But one might also see the growth of new synaptic spines, new synaptic connections between presynaptic elements and postsynaptic dendrites. And this requires the activation of transcriptional regulators. And in this cartoon, we see evidence of a protein kinase pathway activating cyclic AMP and protein kinase A and binding to CREB, which then can regulate the expression of genes that encode for the proteins that are necessary to build new postsynaptic spines to establish new synaptic connections. In this way, synapses can be added following a stimulus that is sufficient to induce long-term potentiation. Now, I would encourage you to review the roles of AMPA receptors and glutamate receptors in long-term potentiation by viewing Animation 8.2. You can click on the hyperlink in your digital copy of the tutorial notes, or you can browse for yourself to the website that supports the textbook that we've been referring to. And I think you'll enjoy seeing the short animation that will help tie together many of these concepts for you. I would also encourage you to consider the study question, which is available at the end of your tutorial, and it's shown here. What I want you to do is I want you to think about why NMDA receptors are so important for synaptic plasticity. And I'll give you a couple of possibilities to think about. So go ahead and answer this question and register your response accordingly. Okay, well, I think we're now ready to move on to consider the opposite form of plasticity that we see in the cerebral cortex, long-term depression. And this too was first studied and its mechanisms well understood within the hippocampus of the rodent model system. So we're going to use the very same Schaefer collateral connection from CA3 to CA1 in order to demonstrate long-term depression. Only now the critical difference is the frequency of stimulation that's applied in order to induce plasticity. Whereas previously we had a short high-frequency burst of action potentials as our stimulus, now what we have is a more extensive period of low-frequency stimulation as our stimulus that induces long-term depression. And indeed, what is seen is when there is a period of low-frequency stimulation, 1 hertz as opposed to 100 hertz or even 500 hertz, what we find is a reduction by nearly 50% in the amplitude of the excitatory postsynaptic potential at that CA3 to CA1 Schaefer collateral synapse. So this is, in fact, long-term depression. Like long-term potentiation, long-term depression can be measured for very long periods of time, beyond the hour or so that is used here in this experiment. So long-term depression can last for days, for weeks, for months, as best as we have observed in our animal model work. We think, like long-term potentiation, this is a very important means of modifying synapses across a lifespan. Well, let's consider the molecular mechanisms of how long-term depression is induced and how it's sustained over time. Long-term depression, like long-term potentiation, require the activity of both the presynaptic element and the postsynaptic process. So there is a requirement for activity in order for the synapse to be depressed. But what's different is the nature of that activity. And I've already suggested what might be different. For long-term depression, there is low-frequency stimulation. That stimulation is sufficient to cause the release of glutamate, of course, and glutamate binds to AMPA receptors and NMDA receptors, and the amount of depolarization that the low-frequency stimulation provides this postsynaptic spine is sufficient to expel magnesium from the pore of the NMDA receptor channel. However, the amount of depolarization that leads to depression is not so great that we have a rapid, high-level increase in postsynaptic calcium. Rather, the amount of calcium that rises within the spine is relatively modest compared to what we saw with the stimulus that gives rise to long-term potentiation. So what we have is a slower, lower level increase in postsynaptic calcium. That is critical. That lower level increase in postsynaptic calcium activates not protein kinases now, but phosphatases. So these protein phosphatases then dephosphorylate target proteins. And that seems to have the opposite effect on the proteins that traffic AMPA receptors between vesicular pools in the postsynaptic spine and the postsynaptic membrane. With the activation of these protein phosphatases, what we see is the internalization of AMPA receptors. So the removal of AMPA receptors from the postsynaptic membrane means that we cannot generate as much current density at that postsynaptic membrane when glutamate is released. And that, in effect, is the weakening of the postsynaptic process. So remember, both long-term potentiation and long-term depression require activity. They both require activation of AMPA receptors and glutamate receptors. The key difference is the level of postsynaptic calcium and the rate of rise of postsynaptic calcium. When calcium rushes into that postsynaptic membrane, then one might anticipate the activation of kinases and the insertion of new AMPA receptors, leading to the potentiation of that synapse. But if the rise of calcium is slow and to a much lesser level, then we expect the activation of protein phosphatases and the internalization of AMPA receptors, thereby depressing the strength of that synaptic connection. Okay, we're going to shift gears now and talk about plasticity in a different part of the brain. Rather than the cerebral cortex, we're going to think about plasticity in the cerebellar cortex. And there, the dominant form of plasticity that seems to shape the function of the circuitry of the cerebellum is long-term depression. And there are some important differences in the details of long-term depression in the cerebellar cortex that I want you to know. And when we come back and talk about the cerebellum, I think this consideration of plasticity in this circuit will make much more sense to you. Now, the cerebellum has some of the most beautiful architecture of any set of neural connections found anywhere in the brain. So I won't possibly attempt to do it justice or to introduce you to all of that right now, but rather so that you can understand the basic players, I'll just highlight a few of them here. The principal neuron that's found in the cortex of the cerebellum, it's not a pyramidal cell, but rather it's called a Purkinje cell. So the Purkinje cell is really just an amazing cell. It has a huge cell body. It grows a beautiful dendritic arbor that has a very interesting geometry. We'll come back and talk about that. And the geometry of this dendrite allows it to receive perhaps as many as 100,000 or more inputs from tiny axons that run through this dendrite. So these axons are called parallel fibers, and as they pass through the dendrites of the Purkinje cells, they make synaptic connections. So the principal synapse in question when we consider long-term depression is the synaptic connection between the parallel fiber and the Purkinje cell. Now, there's another important connection that I need to tell you about that makes plasticity in the cerebellum possible. And it is the connection that comes from a nucleus in the brainstem that has a very nice name. It's called the inferior olivary nucleus. And if you'd looked at the outside of the medulla, you'd see something that we call the olive. And the olive is a bulge in the ventrolateral surface in the medulla caused by the growth and development of this nucleus. Well, this nucleus contains cells that grow out these very special axons that enter the cerebellar cortex and wrap around the primary dendrites of these cerebellar Purkinje cells. And because of this intimate relationship between these growing axons and the Purkinje cell dendrites, these axons are called climbing fibers. Now, these climbing fibers make an incredibly strong synaptic connection with the dendrites of the Purkinje neurons, perhaps the strongest synapse that we know about in the brain. It's right here in the cerebellum between the climbing fiber and the Purkinje cell. So we've got these two synaptic inputs onto the Purkinje cell that we're going to consider and see how their concurrent activation leads to the depression of the connection between the parallel fiber and the Purkinje cell. So again, we're thinking about the cerebellum. Just want to emphasize that point, not the cerebral cortex. So let's see what the phenomenon actually is. So the pairing of activity between the climbing fiber and the parallel fiber is what causes the depression and the strength of the connection between the parallel fiber and the Purkinje cell. So what we're going to measure here is the amplitude of the excitatory postsynaptic potential that is induced by stimulation of the parallel fiber. Now, the test phase is the pairing of the activation of the climbing fiber and the parallel fiber. And what we see is that after that test phase, there is a reduction in the amplitude of the excitatory postsynaptic potential, so that this is now depressed by about 50% following this pairing of climbing fiber and parallel fiber activation. So this is long-term depression in the cerebellar cortex, and it's mediated via the internalization of AMPA receptors. So the synapse between the parallel fiber and the Purkinje cell is a glutamatergic synapse. Glutamate's released, it can interact with receptors for glutamate. Only here, notice that we do not have NMDA receptors, or at least NMDA receptors are not contributing in an important way to this form of cerebellar plasticity. Rather, what we have are metabotropic receptors for glutamate. So these metabotropic receptors, as you may recall, are receptors that activate G proteins that are associated with their cytoplasmic face. And those G proteins then can go on and activate other effector systems within the postsynaptic process. In this case, those effector systems involve phospholipase C and the IP2 pathways. That leads to the generation of IP3, or inositol triphosphate, and diacylglycerol. These second messenger systems can cause the release of calcium from intracellular stores. So calcium is flooding into this postsynaptic spine with the activation of this metabotropic glutamate receptor. Meanwhile, calcium is flooding into this postsynaptic spine because the climbing fiber has been concurrently activated. And as I mentioned, the climbing fiber gives rise to one of the most powerful synaptic connections in all of the brain. This synaptic connection induces significant depolarization into this dendritic tree. That depolarization opens up voltage-gated calcium channels, allowing calcium to flood into the cell. So calcium coming from outside as well as calcium coming from the intracellular stores results in a significant and rapid rise in intracellular calcium in the postsynaptic spine of the Purkinje cell. This sudden and high-level rise in intracellular calcium interacts with calcium-sensitive mechanisms that internalize AMPA receptors. So in this model system, protein kinase C phosphorylates target proteins that lead to the internalization of AMPA receptors. So AMPA receptors are being drawn from the postsynaptic membrane of these spines into vesicular compartments within the postsynaptic process. Removal of AMPA receptors is depressing this synaptic connection. Now, let's be clear. In the cerebral cortex, high levels of calcium lead to potentiation. But in the cerebellar cortex, high levels of calcium leads to depression. And the difference has to do with the second messenger cascades and the consequence of activating those cascades. In the cerebral cortex, those cascades end up inserting AMPA receptors into the postsynaptic membrane. But here in the cerebellum, a different cascade leads to the internalization of AMPA receptors and the depression of that synaptic connection. Now we've come to the conclusion of this tutorial, and we're ready to begin to apply these concepts and think about how synaptic plasticity might play out in the context of ongoing patterns of activity in the brain, rather than those induced by stimulating with microelectrodes in experimental situations. And this brings us to a fairly new discovery in our growing understanding of the mechanisms of synaptic plasticity. So our next tutorial will be about spike timing dependent plasticity. I'll see you then. Welcome to the second of the 3 tutorials that I have for you about synaptic plasticity. In this tutorial, we're going to talk about spike timing dependent plasticity. So again, our concern is for 3 very important core concepts in the field of neuroscience. We continue to talk about the communication among neurons using electrical and chemical signals, and how life experiences can change the structure and function of the nervous system. And as we learn more about these fundamental properties, we are going to learn how better to promote healthy living and the treatment of disease. So again, this is a very exciting field in modern neuroscience, and the future couldn't be brighter in terms of anticipating how discovery in the field of neuroscience is going to improve the human condition. Our learning objectives are, again, to characterize general cellular mechanisms for synaptic change. And then I want you to be able to discuss the importance of the timing of the postsynaptic response relative to the presynaptic activity for the mechanism of synaptic plasticity that we observe in the postsynaptic cell. So those are our learning objectives. And to get started, let's review again the general cellular mechanisms that lead to synaptic plasticity. Well, we went over this in the previous tutorial, and perhaps it bears repetition. Synaptic plasticity begins when neural activity triggers the activation of postsynaptic second messenger systems. The trigger in that postsynaptic cell is typically an alteration in the level of intracellular calcium. The level of intracellular calcium is critical because calcium-dependent second messenger systems are then activated, and those systems can then activate either protein kinases, which phosphorylate target proteins, or protein phosphatases, which dephosphorylate target proteins. And it's the alteration in protein phosphorylation that either turns on or turns off target proteins, and together these events mediate the early stages of long-term synaptic plasticity. So these are mechanisms that are operating upon cellular and molecular machinery already present within the postsynaptic cell. Long-term synaptic change, however, requires alterations in gene transcription. So this is the general scenario by which plasticity plays out, typically in a calcium-dependent manner. And the key difference between one valence of plasticity, if you will, or another is the level of intracellular calcium that's triggered by the synaptic activity. Well, what I want to tell you about today in this tutorial is something called spike timing dependent plasticity. And I want to make clear I'm not talking about a totally different form of plasticity than what we've already discussed in terms of long-term potentiation and long-term depression. Rather, I want to help you understand through this spike timing dependent framework how plasticity plays out in ongoing patterns of activity in the brain. Spike timing dependent plasticity provides for us a framework for understanding plasticity in the brain, one postsynaptic spike at a time. And here's what I mean by that. Real synapses seldom, if ever, experience brief trains of high-frequency stimulation of the sort that were used in the hippocampal slice preparations in order to demonstrate long-term potentiation. So one might reasonably ask, how then does plasticity work in real circuits if they don't experience the kind of stimuli that typically were used, at least in the classical studies of long-term potentiation? Well, here's the spike timing dependent plasticity framework. Again, it allows us to understand these principles of plasticity one spike at a time without necessarily depending upon a special train of stimuli in order to induce the synaptic change. So, consider a synapse where we've got a presynaptic input to a postsynaptic neuron, and in our experiments that I'll be talking about here, it's possible to stimulate the presynaptic axon and induce an action potential at that presynaptic terminal at some period of time, either before or after an action potential is recorded in the postsynaptic neuron. Now, if this is a mature, strong synapse, you might anticipate that activity in the presynaptic terminal will lead to the generation of an action potential in the postsynaptic cell. And indeed, it's possible to find such synapses for which that behavior is observed. And that's what we find in this illustration here in figure 8.18 from your textbook. So there is an action potential in the presynaptic cell, and about 20 milliseconds later, there's an action potential in the postsynaptic cell. So we call this presynaptic activity before postsynaptic activity. This is what we might expect if our presynaptic cell, let's call that neuron A, is firing an action potential, and that leads to the activation of our second cell. We can call that neuron B. So for such a synaptic connection, it's possible to look over time what is the amplitude of the postsynaptic potential or the postsynaptic current following some structured pairing of activity between the pre and the postsynaptic element. And so that's what's being shown here in this recording. We have excitatory postsynaptic current amplitude on the y-axis and time on the x-axis relative to some structured stimulation between the pre and the postsynaptic element. And what we find is that when we provide that stimulus, where the presynaptic input happens before the postsynaptic input, then there is an elevation of the excitatory postsynaptic current. So the amplitude is increased by about 40%, and so this is evidence of long-term potentiation in this pre-before postsynaptic regime. Now, since we can control the activity with our microelectrodes, it's possible to stimulate the presynaptic terminal at a time that actually comes following the generation of a postsynaptic action potential. Now, I'll just ask you to remember that our typical neurons that we have in structures like the cerebral cortex receive inputs from many hundreds, even thousands of different synaptic connections, different axonal inputs converging upon the dendrites of the very same cell. So that cell might fire an action potential in response to activity, and one of these many other hundreds or thousands of inputs. So we're studying one particular synaptic connection, and we're varying the timing at which that input is activated relative to the firing of a postsynaptic action potential. So if we stimulate such that the postsynaptic action potential actually happens before the presynaptic action potential, we might find a recording that looks like this. So the large action potential represents the postsynaptic spike, and then some period of time following it, maybe 30 milliseconds or so later, the presynaptic input is stimulated, and we see some postsynaptic potential that's recorded. But that postsynaptic potential, importantly in this regime, follows the triggering of the action potential. And under that situation, where postsynaptic activity happens before presynaptic activity, notice that there now is the development of a weakening in the amplitude of the excitatory postsynaptic current. This is long-term depression. This is what happens basically when the postsynaptic neuron is being influenced to fire an action potential by some other input other than the one that we're stimulating in this experiment. When experiments such as these can be done over and over again, varying the interval between presynaptic activity and postsynaptic activity, it's possible to generate a function that now accounts for spike timing dependent plasticity. And what we see is that in the pre-before-post regime, that is, when a presynaptic input is in a position to fire the postsynaptic cell, there may be a marked degree of potentiation. So this then is long-term potentiation that can be recorded. If the interval between the presynaptic firing and the postsynaptic firing is less than about 20 milliseconds. Now, 20 milliseconds, it's kind of a magical interval, if you follow me here. That's an interval that's suggestive of a monosynaptic connection where the input in question, the one that we can control with a microelectrode, is effective in triggering the action potential in the postsynaptic cell. We might think of this as as providing some coordination of activity, if you will, between the pre and the postsynaptic element, where the presynaptic input is reliably leading to the activation of that postsynaptic cell. So pre, before, post leads to long-term potentiation. On the other hand, again, recalling that that postsynaptic neuron is going to be receiving hundreds or thousands of other inputs, it may fire an action potential in response to some other input than the one that we have control of with our microelectrode. Well, should that happen before the stimulus is applied to the input in question, we might have long-term depression in this post-before-pre regime. So this now is long-term depression, where the excitatory postsynaptic current is actually diminished when the firing of the postsynaptic cell precedes the firing of the input in question. So we might imagine that this post-before-pre is a picture of incoordination between the pre and postsynaptic elements in question. Well, here in this part of the country, especially as winter is turning into spring, one sport that is popular in my own family anyway is baseball. And baseball involves a pitcher throwing a ball at a batter that has a wooden or a metal bat, and the batter has to time the swing just right in order to hit that baseball. Well, if the batter begins to swing the bat just at the right moment in time, then he or she might strike the ball and might hit a home run. That's a good thing for the batter. We might consider that to be a metaphor for long-term potentiation. If the swing of the bat occurs at just the right time. As the ball is entering the hitting zone, then the batter begins to pass the bat through the hitting zone, and the two make contact, and, well, that's a good thing for the batter, so we'll call that long-term potentiation. Well, there's a particular kind of pitch in the sport of baseball that's intended to deceive the batter. The batter sees the pitcher release the ball, and the batter anticipates that the ball is going at a faster rate than it actually is. This is called a change-up pitch, meaning a change in the speed of the ball. Well, an effective change-up will result in the batter swinging too early. That is, the bat will pass through the hitting zone before the baseball arrives. I like to think of that as post before pre. The bat passes through the hitting zone too soon, before the opportunity to hit the ball arrives. Well, that's not a good thing for the batter. We might call that long-term depression. Well, I was actually invited to think about this metaphor by a student I had a few years ago who thought about this spike timing dependent plasticity function and said, oh, so long-term depression is a near miss. And I thought, exactly, it's a near miss, like swinging through a changeup. It's a near miss. The batter swings the bat, but prematurely, before the arrival of the baseball. The postsynaptic activity happens just before the presynaptic activity. That's actually quite bad for that synaptic connection. That leads to the depression of that connection. Whereas if the batter never swung the bat, if it stayed on his or her shoulder, there'd be no strike. Well, I think that's an important lesson. In order for the synaptic connection to be depressed, it actually has to be used, but it has to be used poorly. That is to say, there has to be some incoordination at that synapse where the postsynaptic neuron fires in response to some other input, not the one in question. But it needs to be a near miss. That postsynaptic neuron needs to fire before, but just before, the presynaptic neuron. Now, I want you to notice something about the shape of this function. Notice that the shape of the long-term potentiation phase is roughly about half the width of the shape of the long-term depression function. Well, I think that's kind of interesting. My background is in developmental neurobiology, so I like to think about how plasticity works out in the context of development. And as neural circuits become active, there seems to be a lot of noise in those activity patterns in the developing brain. And we might think of that noise as being somewhat unstructured and somewhat unshaped by either the brain itself or by its interactions with the outside world. Well, if the interval for depression is twice as large as the interval for potentiation, then that suggests that more of that noise is going to fall in the phase of this function that favors depression, such that the noise is going to tend to lead to the depression of those active synaptic connections. Whereas those synaptic connections that are mediating some structure, where there is presynaptic activity leading to postsynaptic activity, that's coordination. So as signal emerges out of the noise of the developing brain, there's the potential to focus in on that signal, potentiate those synapses that are effectively driving activity in those circuits, and meanwhile depress the other weaker inputs that are out of sync, that are not being coordinated, not being structured. So this may be one mechanism by which the brain imposes a temporal filter on its own activity patterns as a way of selecting those that are leading to an increase in the coordination of firing across synaptic connections. Well, one way that the brain has of imposing some structure over its spatial and temporal patterns of activity is through interactions with the environment. And in the context of a developing brain, it's intriguing to hypothesize that sensory experience might be one source of structure that favors the potentiation of functional synaptic connections, whereas the noise that otherwise is not being well structured by sensory experience might lead to depression and perhaps the weakening and maybe even the loss and the pruning away of synaptic connections that are not functional. Well, this brings us to the close of this tutorial. The third and final tutorial in this series will put all of this together in a very powerful framework that has been with us for many decades now and continues to inspire our understanding of synaptic plasticity. So we'll talk about Hebb's postulate in our next session. Well, now we come to the final of our 3 tutorials on synaptic plasticity, and in this session I'd like to talk to you about Hebb's postulate. Again, we are talking about content that pertains to 3 of our core concepts in the field of neuroscience. We continue to talk about how neurons communicate through electrical and chemical signals, and we're elaborating on a new theme for us, and that is how life experiences can change the structure and function of the nervous system. And as I've commented on in the previous 2 tutorials, I think this is a tremendously important and exciting field within neuroscience, because what we are now capable of discovering are the very mechanisms that we can harness and use to promote healthy living and the treatment of disease and the rehabilitation from neurological dysfunction and disability. Well, my learning objectives for you in this session are, again, to relate our discussion to general cellular mechanisms of synaptic change, and I want you to be able to state Hebb's postulate and discuss its relevance for neuroplasticity. Okay, we've gone through this a few times already, and we'll do it again. Very briefly, just to remind you of our cellular mechanisms of synaptic plasticity. Well, this begins with neural activity triggering the activation of postsynaptic second messenger systems. That trigger is typically an alteration in the level of intracellular calcium that's free in the cytoplasm of the postsynaptic process. That is critically important because the amount of calcium in that intracellular process is going to activate calcium-dependent second messenger systems. And these second messenger systems generally alter 2 categories of enzymes that are present within the postsynaptic cell. Either protein kinases, which tend to turn on target proteins through phosphorylation, or protein phosphatases, that tend to inactivate target proteins through dephosphorylation. So this alteration in protein phosphorylation is very important in mediating the early stages of long-term plasticity, because what is happening here doesn't involve the generation of new proteins, just the alteration in the structural and functional competency of existing proteins. But to get to the long-term stages of synaptic plasticity, there needs to be a means for altering gene expression. So that's the basic scenario by which synaptic plasticity has both short and long-term consequences on the structure and function of neuronal connections. So now I want us to consider a very powerful postulate that's been with us in the field of neuroscience for, well, more than 60 years now. But it's been especially in the last 20 to 30 years that this postulate has really proved to be formative in understanding cellular and synaptic mechanisms of synaptic change. Well, this is a postulate that was proposed by the Canadian psychologist Donald Hebb, who passed away in 1985. And the usual form of this postulate that is often reproduced and quoted is from a very important work of Dr. Hebb's, published in 1949, called The Organization of Behavior. And I know many of you will be interested to learn more about Donald Hebb than I'll have to say. And there's some excellent compilation of information found in Wikipedia. I don't normally recommend Wikipedia, but this is a good starting place for someone just trying to gather some resources concerning the life and the work of Donald Hebb. Well, that brings us to the principles of the postulate. What Hebb's postulate stated, in principle, I'm not going to give you the verbatim text, but in principle, is that coordination of activity, or coordinated activity, of a presynaptic terminal and a postsynaptic cell would lead to the strengthening of that synaptic connection between them. And conversely, uncoordinated activity between synaptic partners would weaken their synaptic connections. Now, a colleague of mine from Duke University who sadly was taken far too early and with great heartache for his family and his friends who knew him well, Dr. Larry Katz. Dr. Katz encapsulated these principles quite insightfully, as he was known to do, in the following phrase: neurons that fire together wire together. The firing together captures beautifully the Hebbian notion of coordinated activity, and the wiring together speaks powerfully about the consequences of coordinated activity for not only synaptic strength, but also the long-term consequences that require the building up of new synaptic connections. Now, Hebb's postulate really is not a departure from what we've been talking about so far in our tutorials on synaptic plasticity. The changes in synaptic strength that Hebb could only imagine in his day, and now we come to speak of in these terms, are long-term potentiation and long-term depression. So LTP and LTD would be the logical consequences of the coordination or incoordination of activity that Hebb postulated. Now, how does spike timing dependent plasticity fit in? Well, again, spike timing dependent plasticity explains how synaptic change can occur one postsynaptic spike at a time under normal physiological conditions that are found in real neural networks with ongoing activity. So spike timing dependent plasticity has been a framework that has been based upon data such as what we've seen previously. Here in this figure, we see that when presynaptic activity comes before postsynaptic activity within a fairly short interval of about 20 milliseconds or so, then we see long-term potentiation. So pre, before, post leads to long-term potentiation. Conversely, if we see postsynaptic activity, presumably driven by some other input to a neuron than the one that we have command of, when that postsynaptic activity comes before presynaptic activity, then that's a regime that favors long-term depression. So now, to put these data in the context of Hebbian plasticity, or Hebb's postulate, we might say that pre, before, post is equivalent to the kind of coordination of firing across a synaptic connection that would be consistent with the strengthening of that synaptic connection. Whereas the post, before, pre regime might be a picture of what incoordination of activity might mean, at least for the synaptic connection under study. Presumably there's coordination at some of the other synapses to this postsynaptic neuron, otherwise it wouldn't be firing an action potential. But in this particular isolated synapse that's under study here, it doesn't appear as though the presynaptic element is really what's driving the activity of that postsynaptic neuron. Hence the incoordination, at least at that one synaptic junction. Okay, so again, Hebb's postulate is captured by this phrase, neurons that fire together, wire together. And understanding plasticity at this level begins to take us from one synapse to the compounding impact across an entire set of connections that builds up neural circuits and even neural systems. So again, change at the synaptic level can produce change in wiring patterns of entire neural circuits. This is why I love this phrase from Larry about wiring together, because it really points to the long-term and broader consequences of a change in activity that occurs at the synaptic level. And so this synaptic change accumulates throughout a network of interconnected neurons, and the net effect of firing or misfiring may be wholesale change in the structure and function of network properties. So in this way, the unitary change of synaptic strength can be compounded throughout an entire network of connections. Well, this is the briefest of our tutorials that I prepared for you so far, but only in terms of my participation with you here. I hope that this tutorial continues in the discussion forum as you consider the study question that I will leave you with, and it's a question that is reproduced for you at the bottom of your tutorial. As I've been mentioning, my former colleague here at Duke, Dr. Larry Katz, proposed the aphorism, neurons that fire together, wire together. Well, this is a wonderful phrase to take away from this tutorial, and perhaps it sounds familiar to an aphorism that you may know. That aphorism is use it or lose it. And this might appear to be a valid restatement of Hebb's postulate, but this is the question I want you to consider, and perhaps even kick it into the discussion forum and share your thoughts about it. So here's my question that I leave you with today. Is use it or lose it an accurate expression that reflects the synaptic mechanisms of neuroplasticity? Maybe you have a definitive yes or no, or maybe you have a definitive no one knows. So go ahead and think about it, and let's see what kind of discussion we can generate around this question.